people come in. All right, I'm going to get started with some housekeeping items. We'd like to thank our extension partners for their support. If you would like to ask a question, you can type your question into the Q&A. Closed captioning is enabled, and you can turn it on and off with the button marked CC or live transcript. I'd like to thank our speakers for presenting and answering questions today, and thank you to Caroline for taking care of logistics for this program. She is going to put a survey into the chat at the end of the program today, but also we'll be sending this survey when we send you the recording, and that'll be in a couple of days, about 48 hours. Thank you for attending today's program. And now I'm going to stop sharing. So that our first speaker can share his presentation. I'm going to give a quick introduction to Brent. Brent, thank you so much for joining us today. Brent Sams has a PhD in horticulture from the University of Adelaide. He has an MS in geography from Virginia Tech and a Bachelor of Science in Geography from the University of North Alabama. He's currently a viticulture research scientist at E&J Gallo Winery. He designs and executes research projects that are focused on understanding how fruit chemistry and quality change over time and space. He's interested in these changes from the within vineyard scale to the regional scale. To accomplish this, he and a team of researchers use a wide variety of field measurements, for example, fruit zone light exposure, vine canopy temperature, soil cores, and many others, also proximal sensing and remote sensing. And he spends a good deal of time analyzing how and whether these measurements can be used to predict outcomes. Welcome to the seminar today, and thank you again so much for giving your talk today titled Spatial Variability in Yield and Fruit Quality. Thanks, Brent.
All right. Thanks for the introduction, and welcome everybody. She mentioned my title was Spatial Variability in Yield and Quality. I know this is a harvest readiness webinar, so I tried to gear this towards like what you should be thinking about in terms of spatial variability and how it relates to harvest logistics, sampling, different things like that. So we will cover a little bit about precision agriculture, yield monitors and mapping, some spatial and temporal variability, impacts on how this impacts vineyard sampling. We'll look at some of what's going on in the yield and fruit quality sensor development area, and then some variable rate management to tie it together. So this is the loop of how precision agriculture works. You take an observation, we evaluate and interpret it, and then we deploy some kind of a targeted management, and then the loop starts over. So this doesn't stop once you have one yield map or one image, something like that. You always want to be looking, evaluating. If you're doing something differently in the field, then you want to make sure you've got an observation that you can compare to previously. So yield mapping is what we look at as the cornerstone for precision agriculture. And this is the combination of everything that's been done in the field by the vineyard manager, by the viticulturist, by the field crews, anybody that's working in the field, plus the impact of climate, topography, soil, all of those different things come together to show you the final result of the yield that was the yield that you got for that particular season. Can be complemented by canopy and soil maps. And so the reason I say complemented is, for instance, if you're doing some of those variable rate practices, particularly within season, you may have a yield map from last year that may look different if you did something different according to to printing or something that you did later on based on that yield map. So you can see sort of changes in canopy size. Soil maps are useful, I think, particularly when they look the same as everything else that you're looking at. So if your imagery looks like your yield map and your yield map looks like the soil map, you can be pretty confident that if you do something differently in that zone or something, that you're going to see some change, or that you have some confidence in your management zones. So yield maps are not all that common. We have a bunch of them running around in our Central Valley vineyards, but this is a place where I think that we could see some movement in the industry. I think it would be really useful for people to start knowing something about spatial variability. But the question that I get most often is, what do I have now that I have a yield map? What do I do now? So the first thing you can start to look at is analyze productivity. So this is just an example using the numbers from the 2021 UC Davis cost study, using the farming cost for a mature vineyard on a single high wire at $600 a ton for San Joaquin County, District 11. So if we've got a yield map, like you see there in the bottom left, total tons, 500 tons, let's give this an average of 10 just to make the math easy. And then, so that would be a total revenue of $300,000. That farming cost comes out to a little bit below 130K, and then net revenue about 170,000. And you see that average net revenue per acre. And again, these are sort of simulated numbers, but that's a decent return. But when you look at the breakdown of the vineyard area, you've got 30% of the vineyard that's either only breaking even or losing money. And so this is where yield mapping and yield monitors can really come in. Most of the units are between $10,000 and $20,000. And so if you know something about your yield, and let's say you can increase yield by a ton or 2 tons per acre, these numbers start to really shift back in the other direction, and that yield monitor could potentially pay for itself relatively quickly. So this is the first place I think that as we talk about harvest readiness, this is one way, yield mapping is one way that you can kind of maximize your harvest efficiency to see more revenue. The other thing we start to look at is spatial structure, or spatiotemporal structure. This is the change in the pattern over time and space. So what we see here is a lot of moving around zones. You've got low yield in 2012 there in the sort of upper middle section. Then it's kind of all the same or pretty relatively close to the same. Now it's the high yield in 2014, and then kind of high-ish yield again in 2015. This is what we refer to as low spatial structure. So the pattern does not change very much, or the pattern does change very much. So it's low spatial structure. High temporal variability, meaning that over time you have a lot of movement going around between the zones. So high yield doesn't mean high yield this year doesn't necessarily mean high yield next year. Compare that with a block that has relatively high spatial structure, meaning that red zone there in kind of the upper section of the upper right section of the block. It's pretty much the low yield every year. You've got some low yield stretching down towards the bottom of that section as well, but generally the low yield is the low yielding section and the high yielding section is the high yielding section. So this is one of those places where you've got consistency over time. You can start thinking about, what should I do differently in this part versus the other part, or in the case of the way that some other folks in the world, particularly in Australia, they've seen some benefits to split harvesting. So you might take a look at the fruit quality in that low yielding section and the high yielding section and say, I can put the low yielding section maybe in one bottle at one price tier, and I can put the low yield or the high yield in another bottle at another price tier. So sampling to account for spatial variability is also a very interesting thing as we come into harvest. So we know that there is a lot of variability in a lot of vineyards. I've yet to find quote unquote homogeneous vineyard. We do need to understand the range of grape quality by block, particularly, I'm a research scientist for one of the largest wineries in the world. And so when we harvest a vineyard, a lot of times that vineyard could go into the same tank. So we want to know what the variability is in that block. We know that some vineyards have very high quality in some sections and very low quality in other sections. And so we want to manage that risk. But the question always comes, if we want to take a single sample or a very small amount of samples in order to not break the bank on sampling, we need to be very, very good at finding where in the block we should go. And this is a vineyard where I think if you take a sample in that green section, if you look at the yield map in the upper left corner, the figure, if you look in the yield map, you're probably going to get a very different result if you take that sample in the green section versus the red section. And you can see that that yield map matches up pretty well with the anthocyanin map, relatively well with polymeric tannins, quercetin, glycosides as well, and a lot of other similarities between these TA and pH as well. So to accomplish a lot of this, we've started thinking about how to use remote sensing for our within season sampling locations. And so this could be, we've done this with all kinds of different sensors, satellites, airplanes, UAVs, you name it, we've done it. And so the remote sensing aspect of this really gets down to, you remember the spatial variability in the blocks I just showed you in a couple slides ago. This is the same vineyard. In the upper left here, we have a high resolution thermal image. So it's canopy temperature with the inner row removed and then smoothed. And then the NDVI from the same sensor in B, there again with the inner row removed. And then as we look into C and D, you've got Sentinel and Landsat. And though there are some differences in the patterns there, some very small differences in some of the regions, you can see that if you're a vineyard manager, it's unlikely that you would do anything differently based on any of these maps. They all kind of tell the same story. You've got kind of a high yield section at the bottom of the block, a high yield section at the top of the block, or high canopy vigor. And then you've got this low vigor sort of towards the center of the block and towards the upper right corner. And so same goes, if you're going to take a sample from this vineyard, any of these images, any of these sources of imagery would tell you more or less the same thing about where to go to take that sample. So what we've done in a couple different ways, and in one case was to simulate how a single transect based on satellite imagery would be able to help us find one single line to walk down in the field to gather a sample for bricks or fruit quality or yield, whatever it is, that would give you the best understanding of the complete block variability. And so this best fit, when I say this, there's a couple of papers, Jim Myers and Justine Vandenheuvel with some of the folks from our lab have published in the last one in the last couple of years and then one about 10 years ago on this method, which takes the 3 most variable contiguous pixels. So this is 3 pixels in a row in a vineyard that represent the maximum amount of variability for any 3 pixels. So in order to test this, so we did several years ago, we had 4 vineyards where we took 125 samples per vineyard spread out throughout the block so that we could make maps of fruit chemistry. And that's what you see there on the right is an interpolated map from those 125 fruit chemistry samples. And you can see that this best fit transect that we've got on there, the red, yellow, and green, they kind of go across the entire variability of the block. So you go through sort of the low bricks, which is kind of the high vigor section, to the high bricks, which is a little bit of a low vigor section. And so this is how we simulated to see how this worked, and we extracted the values from those maps in these 4 vineyards in the Lodi area. You can see the sample distribution there. So there's 125 A, B, C, and D. There's 125 samples per, so that we can make maps of all those things. And so we wanted to see what the sample frequency would look like compared to where that best fit sample was. So that best fit sample in this example in Vineyard B in 2018, the best fit sample was right about 26.5 bricks. And the sample frequency, so this is the histogram, the percentage of samples there. We think about that bell curve, a normal distribution. Our best fit line hits just about right on the peak of where that sample frequency existed. So if we look at this across several vineyards, 11 of the 12 plots, pretty much everything except one, shows that our best fit line using this method was within 1 degree, and most of the time within 0.5 degree bricks of the 125 samples. So even though a couple of those lines shade off of the direct middle a few times, they are typically within 0.5 degree or 1 degree bricks individually. So another thing that we've been working on, this was a big topic last year, and we just got done presenting this work at the European Conference on Precision Agriculture a couple of weeks ago, is using remote sensing for frost estimates. And so this was another thing that we were looking at for how to adjust our yield estimates later on in the season. So we looked at 3 Lodi Delta area vineyards, 3 different varieties: Chardonnay, Pinot Gris, and Pinot Noir. And we had a UAV fly. We developed 3 damage classes, so high damage, medium damage, and low damage, and had some 5-vine reps in each one of the damage classes. And then we took yield components, grapes, chemistry, and then we dissected some buds from a few of these areas as well. I've got these boxes blocking some of these plots, but if you can sort of see beyond them, in a way, the damage classes there, you see that the NDVI in the high damage classes there in red is quite a bit lower than the medium, and then the medium is quite a bit lower than the high. The interesting thing is that the satellite imagery from a couple days before the frost and the satellite imagery after the frost, or the freeze event, I should say, the satellite imagery, we were able to compute a change detection. So we were able to see where the canopy size went backwards, and we could associate that with a freeze event or cold weather damage. The UAV imagery, however, though it was much higher resolution, it was expensive and it had to be contracted. So we weren't able to do that change detection, because unless you're flying UAVs or manned aircraft in sort of suspicion that there might be a freeze event, you're not going to be able to do that change detection. So the nice thing about the satellite imagery is they're always collecting data, and regardless of what's happening, you'd have problems with cloud cover sometimes, but we were able to get a couple of clear images there. So in this case, this is another situation where we really like the satellites, partly because of the cost. Just some assessments of the fruit. So what we really found was that the count clusters on low damage vines stayed high. The count clusters, these are the clusters that were intentionally we intentionally wanted to have come out of the selected locations, the positions from pruning. You can see that the in green there on the far left figure, in green, the count clusters in the low damage areas were much higher, and nearly 100% yield loss in some cases in count clusters for the medium and high damage classes. Our low damage class in Pinot Noir, unfortunately, was machine harvested before we could collect the data from that spot, but I think that we would probably see a relatively similar pattern given the rest of the data. But what was interesting is the lateral berry weights and the lateral clusters made up for a lot of the loss in yield later on. We particularly see that as the lateral berry weights were quite a bit higher than in the high and medium damage zones as opposed to the low damage zones. And then this is that change detection that I mentioned that we could do. So when you've got satellite imagery collecting all the time, you get one image before the freeze event, one image after the freeze event. You do a simple subtraction from one image to the other, and you see where vegetation might have gone backwards. And if vegetation goes backwards, that typically means that a couple of different things have happened. And as somebody that thinks about spatial variability a lot, when I see something that happens across an entire vineyard, I usually think something like mowing, hedging, thinning, something like that causes the entire vineyard canopy to go backwards in the amount of response that you get from the satellites. But when you have these situations, like there's a block kind of in the middle of that picture there, where you've got this big red hole in the middle, or in the top left there in the left figure, another one where you've got this kind of circular pattern in the middle. Management doesn't typically happen in that way. And so when you have odd patterns, this is where I would stand up and take notice. And so what we were able to do just a couple of weeks after this happened was to be able to tell our vineyard scouts and everybody involved on that side of the business to go, these are the vineyards you should check. This vineyard probably had some freeze issues. This vineyard probably didn't, particularly with this situation last year. Anything that had not hit bud break by the second week of April, or was just now coming out, wasn't affected nearly as much as the stuff in those early varieties where the shoots were maybe close to a foot tall. So we've seen this slide before, but one of the things I wanted to highlight on some of these different ways that you can understand this spatial variability is not just by satellites, airplanes, and UAVs, but you also have tractors that are capable of having some of these different implements installed on them. And so I've had a couple of projects over the last few years working with different folks. So this is one with Mason Earls at UC Davis, using this ag kit, which is we're trying to make the most inexpensive bomb-proof sensor we can find, put it on a machine that's already going through the field, and see if we can get our yield estimation and tracking done that way. This thing can take a lot of pictures of a lot of different things. So pest and disease scouting is also something of interest for us, as well as canopy architecture, shoot counts, all those kinds of things. Another one, and this is a little bit sort of farther out there, some grape quality sensing. I've been doing this with Luca Brillante at Fresno State. We're trying to understand spatial variability of fruit color or fruit chemistry non-destructively. So if we drive an ATV with hyperspectral cameras in many different wavelengths, can we understand something about spatial variability in fruit color? We're going to do some more of that with this year. And then just to wrap this up, so one of the other things, I mentioned at the beginning on precision agriculture, variable rate management is where you can really start to, I think, gain some efficiencies in the way that we do things. And so we set up a variable rate spreader trial for fertilizer compost application in this way. The northern block that you see there is so that we can evaluate these treatments statistically. Those are all 1 acre. Each one of those rectangular bars, the yellow or red or green, those are all 1 acre or about 1 acre zones. And we do all of the evaluation based on the yield maps that we get. So we can actually put some statistics to whether or not the treatments work. And then there in the bottom in the south block is a truly variable rate application. So as I said, we've got 56 treatment acres of the 140 total block acres. It's 1 acres apiece. Got a lot of reps, 0, 4, and 8 tons of compost application, and again, yield map validation. Same thing here, though we built this map was based on clusters derived from imageries and from imagery from Sentinel and from 2 soil surveys, and again, yield map validation. So no significant difference between blocks in the first year, and then we're just now into second year, so the compost stuff already been applied. This year we're looking at some other treatments as well, and we'll await the results from yield maps. So again, this is where you want to start thinking about yield maps. Where am I doing really well, and where could I potentially make some more money? And then we come right back to that observation, evaluation, interpretation, and targeted management. So this loop is really the way to start thinking about variable rate management, how to understand spatial variability, and potentially how to use it to your advantage. So in conclusion, an understanding of yield variability is useful for making money, I think. Then remote sensing can aid in identifying these management zones, particularly within season, if you start to do some of this variable rate management. Sampling location and the timing of the sampling are big factors. So there can be a big difference in what you get in one part of the season versus the other part of the season, or one location versus another location. And then there is a lot of change due to seasonality. So I've seen nearly a whole milligram per gram anthocyanins, a whole milligram per gram berry of anthocyanins change from one year to another in the same area of the same vineyard. And so there is this big change in seasonality. But this variability in yield or canopy growth early season can be a pretty good guide. Where you've got small vines early, you're probably likely to have small vines later unless you do something to change that. So to wrap up, many thanks to everybody listed on the slide, different areas, different locations, everybody that's worked on these projects, many people from Gallo. And I can't give a presentation without advertising our internship. So they're fully underway this year, but if you have anybody interested in an internship next year, we'd love to have them apply. And with that, we'll turn it over.
Do we have any questions? Let me see the Q&A. There is nothing in the Q&A at the moment, but if anyone would like to ask a question, please feel free to type your question into the Q&A. Don't all ask at once. All right. Well, yeah. All right. Well, thank you very much. We really appreciate. Here's Leticia. Great. So we really appreciate your taking the time to give this talk today. And good luck with all the measurements that I know you said you're taking later today.
Yep. Thank you. Good luck, guys.
Thank you.
So that's my turn?
Yes.
You guys are fast. All right. So let me share my screen. If I can find it.
And I'm going to do a quick intro, Leticia.
Sure, go ahead.
All right. So our next speaker is Leticia Chacon-Rodriguez, and she is the winemaker and winery manager in the Department of Viticulture Enology here at UC Davis. She has more than 20 years of experience in the California wine industry. She has also worked at Safe Harbor Wine Storage, Treasury Wine Estates, E&J Gallo Winery, Golden State Vintners, which is now the Wine Group, and Delicato Family Vineyards. She has been the co-chair of the Program Development Committee for the Unified Wine and Grape Symposium for several years, and she participates actively on the Spanish track. She's an active member of the American Society for Enology and Viticulture. She's a past president from 2012 to 2013, and just this year was elected to serve on the AACV board for a 3-year term. She holds a Master of Science in Food Science with an enology emphasis from UC Davis and a Bachelor of Science in Chemical Engineering from the Universidad Nacional Autónoma de Mexico in Mexico City. Thank you, Leticia, and welcome. She's going to be speaking about key grape measurements and things to think about.
Thank you for the great introduction. It's a pleasure to be here. All right, so what do we do, right, to get ready for harvest? So we're going to talk about a little bit about the berry composition, the key metrics, kind of like getting ready for harvest, but we're going to talk much more into this area of the berry and the key metrics. And then I'll touch a little bit about how to get ready in the winery based on that. All right, so starting with berry, the berry has obviously different composition components. Water and minerals are the main ones, and some of the examples are like potassium, magnesium, calcium, nitrate, phosphate, sulfur, iron, and other trace minerals. So that composition will change based on the variety, on the vineyard, and within the vineyard, there's variability. We have also carbohydrates, obviously, and the major, obviously, we know is glucose and fructose. The rest are less carbohydrates. There are so many, but in some cases, it's important to measure pentoses, but because those are unfermentable by yeast. So in some locations, some parts of the world, on some varieties are higher on those numbers. So that's sometimes important for the winemaker to measure those, or for the grape grower. On the acids, where we know tartaric and malic are the major acids. And then in some cases, depending, obviously, it's important to measure acetic acid. Acetic acid, as we all know, is could be produced within a cluster when they kind of expand and grow, and we don't even notice, but they are inside the cluster. We cut in half the cluster when we visit the vineyards. It's important to smell and check and make sure that the acetic acid is not present. But if we have a questionable source of grapes, or is part of the contract, acetic acid is another number that could be important to measure, especially historically, if you have a particular vineyard with a problem with acetic acid or high acetic acid, it's important to measure at that point. Other acids are also important, propionic, butyric, etc. Some places also related to some of the traces of any fungicides or herbicides, and also are part of those compounds that we measure. Another area, and I will touch base a little bit more about phenols. So phenolic acids like hydrocyannamides and hydrobenzoids are one of the major acids. Then we have flavonoids, we have the tannin polymers. We could spend a whole day talking about phenols, because phenols obviously are of particular importance on red grapes, but it's also important, they are also important on the whites when we are looking at the color, the final color, we're looking at the texture, we're looking at the whole mouthfeel, and also what decisions we're going to be making with press fractions and such. So the phenols will be important no matter what, also for the rosés, for the color and the color stability. Nitrogen compounds. So nitrogen compounds, we're talking ammonium salts, amino acids, peptide proteins, and nucleic acid derivatives. The nitrogen compounds are obviously as important for winemakers to make sure that we need to supplement with any nutrients. And I'm sure later on today, you will have a little bit more information about the yeast requirements for nutrients. We don't want to be below the requirements, and we don't want to go above the requirements. Terpenoids. So those are the volatile and aromatic, especially, for example, Muscat varieties. So we have more monoterpenes, which are 10 carbon compounds. We have many other terpenoids that are going to be part of that varietal characteristics. And I skip the presence, but the presence can be into nitrogen, it could be into, depending how they tied in, it could be volatile aromas. They can be classified under the different very components, depending the properties. But purines, for example, like Cabernet Sauvignon, Sauvignon Blanc, they are mainly the compound that gives you like the bell pepper characteristics. So for winemakers, is part of the characteristical varietal. When we just refer to Cabernet Sauvignon, for example, we're like, okay, it has to have a little bit of these purines, but if to what level, to what degree, and how much is this balance with the other fruity aromas, that's when the decision of the winemaker comes, and also working together with the grape grower to make sure that we achieve the consumer profile that we're looking for that particular variety. And then obviously we have lipids and related compounds, which are mainly found on the seeds, also in the exterior of the skin. And then we have volatile aroma compounds, which are very characteristic for each of the varieties. And that's where the winemakers, we just like spend a lot more time talking about. So what are the main challenges when we are checking the berries and when we're going in the vineyard? So first of all, it's like, what is the vineyard sampling? How large is the vineyard? How much is the variability within the vineyard? So there's so many right now with AI and then GPS and all these pictures from the sky, we can see the variability in the vineyard. So it is recommended to go into and identify and get to know that vineyard very well to make sure that we sample each location that are significantly different. And then for the parts that are significantly different, see how large it is and how large my sample needs to be. In here in Davis, obviously, we vary a lot into like a very small vineyard on larger vineyards. So we have small lots, and then we just do grape berries sampling. When we cannot do grape sample, berry sample, we do clusters, right? So it depends how large it is your vineyard and how much of that you can use for. So we have also to be in touch with our grape growers regarding the vineyard practices, in particular talking about irrigation. When we do irrigation, we do have these obviously changes on the composition of the berry. So we need to be able to like, okay, oh, my bricks now is different than a week ago or 3 days ago. We'll continue monitoring, and then all of a sudden we have a dilution factor because of the irrigation. So irrigation and any other practices that are performed in the vineyard are going to be important into how this works. We also have sample preparation, which can indicate, okay, how much do you press? Are you releasing just the, what we'll call like free run, when we are squeezing those berries in the lab? To what pressure are we applying? Are we getting any of that kind of hard press or moderate press composition? Because that changes. And how long it takes between I prepare this sample and I really run the analysis. So there's some oxidation that can interfere with some of the components that sometimes people are interested in too. So, and then another challenge is the distribution of the berry components, which kind of I touch base when we talk about the sample preparation. But obviously, the free kind of press use, or when we squeezing a grape in the vineyard and put it in the refractometer, normally it's about 1 brix higher than what it really is, and it's often half of the acidity compared to the juice, which is near the seeds. So all this distribution of the berry components will make it more challenging when we are preparing samples. So what is the recommendation here is to use the same procedure from sampling, from getting together with your grape grower, to all the preparation of your sample. So all that, if you do it in a very standard procedure, you make procedures, you validate them, you check, and you make sure that that is the procedure that works for you, or if it needs to be updated. So obviously the potassium will be higher when you press higher, the pH also increases, and nitrogen will be higher, and the sugar will decrease, unless you have sugar or raisins in your berries. So this is very important to consider and understand those challenges to really develop a procedure that makes sense for you. And obviously the numbers or the analysis are going to be as good as the procedure and the sampling they're going to have. So the grape cluster composition, so also we need to identify when we are doing cluster sampling, we know that we have stems, we have skins, we have seeds, we have the fresh pulp and the juice. Kind of this is the percentage depending on the variety, depending on the vineyard. So smaller berries will have obviously more skin than other larger berries. So here's a little bit of the phenolics that are of interest for the winemakers. We talk about phenolic acid, flavonoids, anthocyanins, which are related to the color. So the anthocyanins are directly the color of the grapes. And then we have tannins, and we have polymeric pigment. So more or less this distribution you can see in here. And this one is also a good picture to identify how do we taste the grapes, because we also want to taste them. So when we go out there, we want to separate the skin, where we'll have more anthocyanins, proanthocyanins, and flavor compounds, and taste them separated from the flesh, from the pulp, which the flesh and the pulp will be mainly water, some organic acid, and sugar, with some flavor compounds. And then observe the seeds. Obviously, we don't crunch on the seeds. We don't, because then you will get distracted on your mouth. You won't be able to taste anymore, because you will be old with the bitterness and the astringency on your mouth. So the seeds, it's important to look at the color. How much is the separation from the seeds from the pulp and the seeds? The more ripe, depending the variety, is normally the seeds will turn browner, from green to brown, and it will be released easier. And that will determine also how much are you going to be pressing how much are you going to be doing those pressing separations when you decide to bring the fruit into the cellar. So some of the phenolics that we have are, like, as I said, the seeds and the skins. We other components of phenolics, and the winemaking world is coming from the oak and some microbes. Those are here just the typical concentrations, and this is the red wine composition without water and ethanol, which is related to, obviously, to the grapes. So what we're trying to do on the grapes measurements is to identify how much of that grape is going to be translated into your red wine composition. So like kind of summarizing a little bit of the key berry metrics, we're talking about, obviously, sugar, we're talking about acidity, tartaric acid, malic acid, and the tetrable total acidity. So if we want to identify the individuals, malic acid will be important, especially if we're going to be doing the malolactic fermentation. Plus also, if this concentration is higher than you want to, it will change the profile of your wine. So depending the consumer profile or the wine style that you want to produce, you want to pay attention important to the malic acid. Obviously, the weight, how much or the yields are we getting, how heavy, how we're going to be producing this. Anthocyanins and total phenols that we talk, taste that I mentioned. You want to develop a taste scorecard with your teams to be able to identify and do it in the same way. So then you have a good records on the taste of the different parts of your vineyard. We have vineyard conditions, we have weather conditions to consider. The vineyard conditions, for example, let's say we're going to have a rain tomorrow, and this vineyard floods easily. So if you're going to have a lot of rain, you won't be able to put machinery in here or people in here if it's flooded. So you want to consider that into your harvest decisions. So what else do we consider? Okay, the logistics as well. Do we have enough trucks to get this fruit? Do we have, if it's a small, you have micro beans, you have the people, do you have the crews to do all these? How hard it's going to be? What time do you want to start your harvest? And other risk factors such as smoke, right? Especially like in California, we learned that many years ago and several years ago, or a few years ago, that smoke can impact our vineyards and can affect the whole harvest. So what are the risks? What is happening in the weather? What is happening in the vineyard? And what are my logistics? What are my chances to be more successful? So it's all considering all this picture. So what is important to do in your site? So you want to develop a specific harvest plan. You want to know what the goals are, or your company, or if you're just a home winemaker, for example, what are your particular personal goals? We know that we have large, medium, and small wineries. This is all relative because sometimes you think it's a large winery, but sometimes it's a medium compared to another larger winery. So all this kind of relative, but we kind of have an idea how large we are, how much we have to produce, and what is my timeline. Are we going to do custom crush, or are we going to do private label? All those factors are important to consider in our site-specific plan. Now, what is the most important part of harvest is your team, your groups, your people. You have the winemakers, your grape growers, you have operations and production, you have quality control, your lab team, supply chain, research, and many others. It depends how large your company is or your site is, but people are the most important resource that you have in any operations. So what we want to do is support the teams. Obviously, you have training to support these teams, the food safety training. Remember that now, obviously, wine is a food, it's under FDA compliance. So we want to make sure that food safety training is provided. Winery safety, to make sure everybody comes in one piece and goes home in one piece. That's critical. And make sure that we don't give this for granted. Sometimes we say, okay, we've been here forever. We know we've heard this before. Well, accidents happen exactly when that attitude becomes, when you are too comfortable and you think you know, is when things happen. So safety is always something to be aware of and to refresh it. You have your operating equipment and lab, your instrumentation training. And then you have support teams like compliance. Not everybody has that. Sometimes winemakers are compliance, we're accounting, we're administrative. We have marketing team, sales team, human resources. Those support teams can help you, and it depends, obviously, the structure of the size of your team. Now, standard operation procedures, SOPs, are going to be your best ally on making sure you have a good training, and then you have the same procedures. And then don't just stick with what you have, try to update them as frequent as needed. Sometimes the SOPs are like very old, and then you're like, okay, things have changed, equipment has changed. You have to adapt to that as well. So make sure that you consider that. Logistics and communication, this is a big piece as well. So we want to make sure that we know how the vineyard is going, to get very good information from the vineyard managers, the grape growers. What the challenges are they having? Do we have like a sunburning, or do we have too much? Are there cold or wet conditions or hot conditions? So depending where you are, you want to make sure that you meet with your teams as needed, and then make sure that you have flexible or extended hours as required. But plan ahead so things are not as a surprise as sometimes happens during harvest. Harvest is obviously chaotic. We have to be flexible. We have to make sure we are in our mindset to be flexible, but we want to also plan as much as we can and to control as much as we can control. What other requirements we need? If you are developing a new product, when you have some clients that they want to visit you, you have research to conduct, you have students. What equipment do you have? How you've been making sure that equipment is distributed, is in the site, and is working correctly. You have crusher, fermenters, presses, tanks, centrifuge, filters, etc. What other operations are happening during harvest? And obviously you have the winemaking protocols and the challenges. So on the equipment, so obviously you have bulk wine inventory as well. This is a big deal, especially in larger wineries, to make room for the new vintage. So everybody has that, obviously, but it could be less or more depending your cycle or depending your site. But are you going to be bottling everything to deplete and make room for it? Or are you going to ship to other storage locations? You're going to ship to other locations. You don't want to start the day before you receive grapes. You want to make sure you receive, you start shipping way before you plan for harvest. Preventing maintenance to your crush equipment obviously is important. Make sure it's working, test your equipment, validate your procedures. And then cleaning and organization of the winery housekeeping is going to save you time, and it's going to make you more efficient. We also have research cellar inventory. If here, for example, in Davis, we normally have different projects, and we have to organize them ahead of the game. We want to make sure that we have all the resources for those research projects. And this is just an example of your plan A versus your actual, and you can compare, okay, how much you have, how many presses, juice and tank A and B, what is going to happen to it, flotation. So there are like different ways of organizing per week your capacity, your equipment, and that will be tied up with your groups of different teams of the crew, the people, the labor that is going to be supporting those operations. Your winemaking protocols, obviously you want to make sure the size and the number of fermenters are right on. If you need to empty a fermenter to receive more grapes another day, you may have to call it for pressing before you wanted to. And so then at that point, you have to think, what is the best outcome? Do you want to wait with the fruit out there? Or do you want to press early to make room for that fruit? So that will be obviously part of your day-to-day decisions. What is the budget for each program, which can be linked? There are many databases that help accounting, winemakers, grape growers about how to link the ingredients and the treatment to each program. Let's say that you have program wine A and wine B, or it could be a different varieties, which could be associated with different cost. What is the cost of the grapes? What the cost of everything that you're going to be doing out to the grapes? What the procedures is you're going to do extra, for example, on a rack and return for red fermenter? Do you have that extra capacity to go to another tank and then return? Or you are compromising your fermenters and then you can no longer do that, which it happens, especially when you are in the peak of fermentation and harvest. Then how soon do you need to bottle your program? So do you want to do an early release, or you're going to put it on barrels immediately, and then in barrels you're going to age for 2-3 years before you release it, or you're going to bottle and age on bottle 2 years, 3 years, or just 5 months or 6 weeks. So it's all depending what your program is going to be, obviously how you're going to treat a wine in your winery. You have to consider stabilization, blending, and aging as part of your logistics and your procedures. So how soon you need to put this out in the market. Well, the challenges, obviously the weather, we talk about these, heat, cold waves, rain, vineyard, fires, earthquakes, power outage. I've been in places where we have power outages, and then it's a big challenge when you have, where at the peak of fermentation, you can lose your fermentation. You can lose tanks of must of wine that is in the middle of it. Then we have materials and ingredients. You want to make sure, obviously, you have your inventory on hand, the yeast, your enzymes, your bacteria. You're going to conduct malolactic fermentation, what nutrients, your acid, your oak. You're going to do macro-oxygenation, what the protocol, equipment, what analysis you're going to be doing, how often you need to taste this. You want to make sure you have cleaning and sanitizing agents, your lab reagents, and any other special projects or research projects. So this is just a quick kind of chart of the most important analysis or metrics that we do recommend or we perform. This is the different steps of the process. Those are the basic analysis and the recommended frequency. And then we have other, and there may be much more than that. So in the vineyard, when we have the grapes, we do Brix, pH-TA, sometimes color, phenolics, glucose, fructose, malic acid, volatile phenols, other microbial analyses, ethyl acetate, acetic acid, and such. In the juice, once it arrives to the winery, we do have like Brix, pH-TA, malic, NOPA, ammonia, and free SO2. Those are kind of like the basic analysis and juices and must, and it's done when you receive and crush and press the juices. And then other analysis at this point will be color, phenolics, volatile phenols, etc. Obviously, you want to taste in every step. During alcoholic fermentation, Brix and temp are the classical ones. There's more. Now we have, and it's not here, but it's redox because it's not too common. It's something that we're doing here in Davis. We check redox during alcoholic fermentation to keep it and prevent it from reduction. And this is the Brix and the temperature is recommended once or twice a day. Now there are devices that can check it almost continuously. We do check it 96 times a day, meaning every 15 minutes. So because we have the gadgets to do that, right? So it's just a matter of how important is this. And then now during alcoholic fermentation, we're able to even predict if that one is gonna stick or not. It's gonna be sluggish or not fermented. And I think after me, another person is going to talk a little bit more about that. So I'm just going to go through that pretty quickly. After alcoholic fermentation, once you have your wine produced, you have ethanol, pH, TA, malic acid, glucose, fructose to see if it's dry or not, free SO2, volatile acidity. Once post-alcoholic fermentation, this is what we normally do. Sometimes at this point, we either say, okay, it's not going to go through malo. Then at this point, we're just going to finish one. But if you're going to do malolactic fermentation, it's recommended to do malic acid and volatile acidity every 2 weeks. Sometimes it's every 1 week, especially if you're seeing good numbers, then every week. And other components are resveratrol. Finished wine, you want to have your whole set of ethanol, pH, TA, malic, glucose, fructose, free and total, VA, and your stability, heat and cold stability in the case of rosés and whites. Once after fermentation and blending is when we do this. And then those are other common analysis that we do. And here we're adding, for example, dissolved oxygen to make sure that we are not oxidizing at that point our wines. So this is just a quick summary of the recommendations. So are we ready for harvest? So obviously you want to have like a checklist for harvest readiness. You want to keep your teams informed. You want to always expect the unexpected. What if, right? What happens if my crusher breaks? What happens if my crew gets sick and then I have no crew? What happens if, right? So we have lived through so many challenges so far. And so we are even more prepared than ever for all those unexpected situations. Keep growing, learning after harvest. It's important that every time when you finish harvest, you meet with your teams, meet again, and try to revise all your protocols, everything, and see what can we do better. And then a key thing is always keep a positive attitude. With that, I want to thank you, everybody. And I think there's a question here. Oh, besides a refractometer, is there one tool or gadget that you recommend for collecting key grape measurements for a small winery, not too expensive? Okay, so that was from Chris. So the refractometer is kind of just your sugar at the beginning on the juice, and that can help you with just monitoring how is that going. Acidity will be your other important measurement. There's nowadays other analytical instruments that are not too expensive and they can help you with acidity. So hopefully that helps. Nowadays, I think we have a lot more gadgets than before, and that will be part of what you can use in your site. You can also be together, like get together with other wineries that are in your neighborhood, and maybe all together can build like a small lab with the most basics analysis that you require, like pH, TA. pH is fairly simple, it's an electrode, and you can calibrate very easy. You can have just acidity with pH meter, and that your sensory attributes will be your best ally, obviously there. But you can start investing and getting together with everybody around you, investing in having some equipment is going to pay off in the long term, because you will have more numbers and you will have much more information to even like get more successful or understand problems that you might have in the winery. Like, okay, why did this happen? If you have no numbers, if you don't have information, it will be very hard for you to find out the root cause of that and to really attack that problem. So I think I would recommend you to probably get together and with other wineries that are small and have a little kind of lab, a cooperative lab kind of thing. Any other questions?
Thank you very much, Leticia. It looks like there are no other questions.
All right. Very good.
Thank you so much.
Thank you.
All right, looks like we're still right on time. Our next presenter, as he gets ready to share his screen and his slides, I will do the introduction. So our next presenter is Professor David Block. He's the chair of and professor in the Department of Viticulture Enology at UC Davis. He's also a professor in the Department of Chemical Engineering. Since joining UC Davis, he has conducted research on various topics, from fermentation optimization methods to metabolic engineering of yeast for improved wine production, as well as on single plant resolution, irrigation, sensing, and control. More recently, within the past, I'd say 5 to 6 years, his research is focused on computational fluid dynamics models for red wine fermentations and process optimization for cultivated meat production. He played a key role in designing the UC Davis LEED Platinum Certified Teaching and Research Winery. He's received the Distinguished Teaching Award from the UC Davis Academic Senate, and he is an AICAG Fellow. That's the American Institute of Chemical Engineers. Prior to joining UC Davis, he worked for Hoffman LaRoche, working on biopharmaceutical production, both in process development and in manufacturing. He holds a Bachelor of Science in Engineering from the University of Pennsylvania and a PhD from the University of Minnesota, also in chemical engineering. Thank you for joining us today, Dave, and he's going to be talking about planning for successful fermentations.
Great, thanks, Karen. What Karen didn't say is I just came home from a trip with COVID, so I'm sitting here in bed giving this talk, so hopefully we'll make it all the way through.
And we really appreciate that you are here with us today.
Yeah, so what I'm going to be talking about is planning for successful fermentations. And what I'd like to do is kind of separate this into 2 parts. First of all, we'll talk about fermentation kinetics and how to make sure that we're going to have a successful fermentation in a reasonable amount of time. We're going to go through juice composition, especially sugar and nitrogen, but obviously, as you heard in Leticia's talk, you also have to think about things like SO2 and acid and pH and things like that. We're going to talk about fermentation temperature because that's really critical to having a successful fermentation. We'll talk about yeast characteristics a little bit, but you're going to have a whole talk right after me from Nichola on yeast characteristics. So I won't spend a huge amount of time talking about that. And then very briefly about mixing in white wine fermentations, especially how it relates to getting fermentation started, especially if you're going to start in a tank and go to barrels to complete a white fermentation. And then we're going to switch gears and talk about phenolic extraction red wine fermentations and the considerations there from the effects of temperature, cap management, and scale. So let's talk with fermentation kinetics. We're going to start with juice composition. Many years ago, we came up with a model for how cells grow in a wine fermentation. And essentially, the limiting nutrient, what runs out first is most of the time is nitrogen. And so if you, let's say, double or triple the amount of nitrogen, then you're going to double or triple the amount of cells that you get, which is going to double or triple the rate at which the cells move through metabolize sugar and create ethanol and carbon dioxide. And if you have a lot of cells, they can move through the sugar very quickly, and they spend less time in ethanol, which can deactivate the cells. And so I want to minimize the time that they spend in a lot of ethanol. So we actually turn this into a mathematical model, but it doesn't really matter for these purposes. I want to just go through some of the important findings from that work to think about as you're planning your fermentations for this year. So first of all, increasing sugar increases the risk of stuck fermentations. I think we all know that. This is an example here in this graph where we basically have the same amount of nitrogen. These are flask fermentations. We have the same amount of nitrogen, but with increasing levels of initial sugar. And what you can see is you go from normal fermentations at the 3 lower sugar, and then you sugar levels, 3 or 4, I guess 3 levels. And then as you get to the higher levels of sugar, when you get to 260 grams per liter, around 26 Brix, or 30 around 30 bricks, you start to have problems with fermentation completing. And this is really important because as you're planning when to harvest, as Leticia talked about, you don't want to create problems for yourself later on. Now, of course, if your grapes are dehydrated in the process prior to harvesting them, then you can water back, and that may be a good idea if you think you're going to have a problem with your fermentation completing. So the other thing that I want to talk about is the impact of nitrogen on biomass. So this is actual data along with the model prediction. And what we show here is that as you increase the initial nitrogen, you get more and more cells. Initially, it's proportional. So if you were to quadruple the amount of nitrogen, you'd have quadruple the number of cells. What we can see here is you go from 140 milligrams per liter to 230. You aren't really increasing the cell concentration, or excuse me, probably because something else becomes limiting, either something other than nitrogen or a very specific component of nitrogen. So that means as you go from high nitrogen, where you get a successful fermentation that finishes, and you decrease the initial nitrogen, you go from a normal fermentation to a sluggish fermentation here to a stuck fermentation. And obviously this is something we want to avoid because we're going to end up with residual sugar and probably acetic acid problems and things like that. So we need to have enough nitrogen in that fermentation at the beginning. And there are different numbers out there. And I think maybe Nichola will talk about that a little bit because it may be based on the yeast that you use. Certainly more than 140 in our studies, milligrams of nitrogen per liter usually led to a normal fermentation, but certainly having more in the order of 200 and 250 milligrams per liter might be safer. What you don't want to do is you don't want to end up having excess nitrogen, because if you do, you have more chance for instability in your wine later on. So one of the things that you might think about, I think you think about in a fermentation, is when do you add the nitrogen? Can you add it later in the fermentation? And this is some simulations we did, mathematical modeling, looking at adding DAP, so diammonium phosphate, at different times in the fermentation. What you can see is the prediction is that when you add it, you get an immediate hike up in total biomass, so you get cell growth, and a faster fermentation. And so what you're seeing is that the prediction is that as you add DAP, you're going to complete a fermentation that would otherwise stick. And what you can see at the bottom is that when you add the nitrogen at various times, the prediction is that it's going to be completely used up almost immediately. So this is data from Julie Schreiber in her master's thesis. And what you can see is the data does actually look very much like this, with one exception. So you can see that you do get these hikes up in cell concentration after you add the DAP. But probably most importantly, you'll see that the DAP is used at less than 200 hours if you add it. By the time you get to about 360 hours, which is about, let's say, 80% through your fermentation, sugar-wise, the nitrogen's no longer taken up by the cells, probably because the cell membrane changes. And so at that point, it's not going to be an effective way to finish a fermentation that might otherwise stick. So you want to add nitrogen earlier rather than later. The form of nitrogen also makes a difference. So that was with DAP. We also did similar kind of thing with superfood, which is a complex source of nitrogen. And what you can see here is in this case, you really do you see this bump of cell concentration as soon as you add the superfood, and these are all added at the same concentration. And interestingly, what you see is even out of 360 hours, you do get some take up of that nitrogen. Now, it's not complete, it's not perfect, but you do get some nitrogen taken up. And so if you're planning to add later in the fermentation for whatever reason, you're starting to sense a problem, complex nitrogen source is going to work better. So just some lessons learned on this. High sugar is going to make other factors more critical. You need enough nitrogen to give you a reasonable fermentation speed, but not too much so you have a lot of extra at the end, which can lead to stability problems. You can supplement nitrogen during fermentation until it isn't taken up anymore, and that's going to happen earlier for DAP than for complex forms of nitrogen, which are going to generally work better than DAP, especially later in the fermentation. Another key factor in successful fermentation is fermentation temperature. And so this is work that was done by Matt Coleman and Russ Fish in my lab a number of years ago. And we did basically the same juice with the same amount of nitrogen at different temperatures from 11 degrees Celsius, which is just over 50 degrees Fahrenheit, to 35 degrees Celsius, which is in the mid 90s Fahrenheit. And what you can see is at the colder temperatures, you have a very slow fermentation, almost sluggish. As you increase the temperature, you get faster and faster until you get a normal fermentation in the 20 to 30, at least 20, 15, 20, 25 degree range. But what's interesting is when you get to higher temperatures, you get very fast fermentations that then stick. So that's something to think about as you're choosing your fermentation temperatures. The other thing is that if your nitrogen level is low, what we found is that your successful limits of temperature range is smaller. So the lower your nitrogen, the smaller range that will work. You can't get too cold or too hot, or you won't have a successful fermentation. So lessons learned on fermentation temperature. Fermentations go from sluggish to normal to stuck as fermentation temperatures increased. Temperature is even more important with low initial nitrogen. Everything speeds up with high temperature. But unfortunately, I didn't go into this in detail, inactivation of your cells speeds up the most. So you're going to lose a lot of the activity of your cells at higher temperature. Just a quick note on mixing. This is work that we did right after I got to Davis. And what we did was we took a fermentation in a 500-gallon fermenter in a tank that actually one of our classes was working on, and they had inoculated the fermenter. It was a white fermentation. And we used a spectrophotometer on a stick, and we just moved it down at 1 foot intervals. And we took the optical density, which should be proportional to the yeast concentration. And what we found was that within the first day or so, all of the yeast settled out. And then it started growing from the bottom of the fermentation. That's the white circles here. And then we actually got more growth here by 2 days. And interestingly, it was lower in the middle, but some had floated off to the top. By 3 days, we had got more growth in the fermenter, but still not much at the top. And it wasn't until 4 days in that we got uniform growth throughout. Now, there's some mitigating factors here. We probably ran this too cold. One thing I will say is that I'm guessing that the students didn't hydrate the yeast thoroughly. So that's really important to make sure you hydrate your yeast thoroughly, or you will get something like this. And where it's a real problem is, let's say you're going to start out for 3 or 4 days in a fermenter and then go into barrel. Some of your barrels, if you were to empty into barrels at 3 days, some of your barrels would be getting significantly less inoculum than the other barrels. And that's going to be a problem in managing those fermentations. So make sure that your fermentations are fully mixed before you go from tank to barrel. Yeast strain differences. All I'll say here, because you're going to hear a much more detailed talk from Nichola, is certainly yeast strains with the same nutrient environment will exhibit very different fermentation characteristics. They'll grow to different cell densities, as you see here with 3 of these. They'll have different Brix curves. This is data for a bunch of different yeast that we looked at, a bunch of commercial yeast. And in the same media, they grow to probably a factor of 2 difference in cell concentration, which will definitely affect the growth, the Brix curve, the rate of maximum sugar utilization. And so that's really important. They'll also reach different ethanol levels. They have different alcohol tolerances. And so really know your yeast, I think, is one way of putting it there. And again, you'll hear more from Nichola on yeast. All right, now I'd like to switch gears and talk a little bit about phenolic extraction red wine. Talking about trying to get the phenolic profile that you're shooting for in your wine, in your red wine. Of course, the problem here is that phenolic extraction in red wines is complicated. You have the cap at the top where a lot of your phenolics are originally. You don't really have any heat transfer from the cap into the jacket. So your cap is going to heat up until you do pump overs or other cap management. You will have pretty good mixing in the liquid below and good temperature control. If your temperature sensor is at the bottom here, you may never know that your cap temperature is getting significantly higher, as much as 30 degrees Fahrenheit higher, we've noticed up here. So the question is, what are the important factors for determining phenolic extraction in red wine? Oops. So we did, this is with Anita and her group, we did a bunch of experiments in our TJ fermenters where we could control the temperature of the cap with pump overs, the temperature of the liquid with the jacket individually. And I'm not going to go into the details of the study, I'll just focus on the results. So these 4 graphs summarize the results. And here I have malvidin 3-glucoside or catechin, but we did this for tannins and other, we measured a full phenolic profile. This just illustrates the point. If we look at malvidin 3-glucoside, which is going to be in the skins of seeds, I'm sorry, skins of the grapes, grape berries, what you can see is the purple here is 35 degrees Celsius. The red is 25 degrees Celsius. This is in the cap and the liquid below. The blue is 20. And I guess missed the red. The red is 25. So what you can see here is they all, regardless of the temperature, reach about the same maximum level. One of the key things here that you can see is at 35 degrees Celsius, you start to lose anthocyanin, and we think this is from degradation, not reaction at this point in the fermentation. So the lesson learned there is skin extraction happens early. It's in the first couple days of the fermentation, and its extent is not temperature dependent. You know, how quickly it gets to that maximum is temperature dependent, but for the most part, it's not going to be temperature dependent. Whoops. So if we look at catechin, which is one indicator of what's happening from seed extraction, then what you can see is that is definitely happening later in the fermentation. Most of our skin extraction happens by here. You can see catechin extraction continues to happen, and it's definitely temperature dependent. the extent and the rate are temperature dependent. So seed extraction happens later, and it's definitely temperature dependent. One of the things that we did find is that, at least in the TJ fermenters, and we'll talk more about this, the phenolic extraction was not a function of the pump over volume or the frequency. And in fact, we did a number of experiments on this, including one experiment where we didn't pump over at all, and you got a somewhat lower extraction of malvidin-3 glucoside, but not that much smaller. And it's interesting, when I shared this result with some winemakers the first time, I had one person who asked me a question. They said, well, we've done this experiment by mistake with an automated pump that wasn't working, and the wine came out pink. Why is that? And so I tried to come up with, ideas of why that was. Some of them were close, some of them were not. But interestingly, as soon as I sat down from giving that talk, I had another winemaker from the same company who came over to me and said, Dave, we've done that experiment by accident, and we got complete color extraction without pumping over at all. So I don't really understand what's going on. So one of the things that we did to try and understand this more is to come up with a mathematical model for extraction to try and see, how extraction, to predict how extraction would happen, not only with different pump over regimes, but different scales and shapes of fermenters and things like that. So I'm not going to go into a lot of the details of this model, but just to say there's a yeast growth model that goes into this. That's the one that we used earlier. I used earlier in those fermentation predictions I showed you. Then there's all the physics. This is the mass transfer, the fluid flow, and the heat transfer. And then there's the properties of materials like the thermal conductivity of pumice, which we had to measure. And you put all this together, and you can predict the spatial distribution of temperature and ethanol in a fermenter. So this is what it looks like. So this is a cross section of a fermenter. You can see right before the pump over, this is basically where the cap is. And you can see the cap is much, much hotter than the liquid below, but the liquid below is pretty uniform. You get good mixing in the liquid below. It's cooled by the jacket, but no cooling by the jacket of the cap. And then when you do a pump over, you get a distribution of heat. So you push the heat down throughout, and then the heat builds up again in the cap. So that's the prediction. So the question is, how good is the prediction? And so this is actual data that we took in a 500-gallon tank. And it's not perfect, but it's pretty good. So it can be pretty certain that the predictions are not going to be that far off for temperature and ethanol. We use this model to look at what happens when you have short squat tanks, short wide tanks, or tall skinny tanks. And you can see these are for different sizes. So these are for 500-liter tanks up to 5,000-liter tanks. And what you can start to see with the 5,000-liter tank is we're supposed to be controlling the temperature at this blue color, and we're not, right? So at some level, and this is 500,000 liters, it's almost impossible to take the heat away, and we're going to need to do something else if we want to cool that tank, probably use an external heat exchanger. All right, so once we were able to predict spatial distribution of temperature and ethanol, then we can develop a model for phenolic extraction. And that model, I'm not going to go into the details, but the model has to do with the release of phenolics from the skins or seeds, the reaction of those phenolics, and then the absorption of them back onto the skins and seeds, or desorption. And these are all critical parts. So Conrad Miller did this work in my lab, and he took this model where all those parts are a function of temperature and ethanol, and you could combine those models into an integrated model to predict phenolic extraction. So to test this out, we had data that we collected. This is from a paper by Larry Lerno with Anita. And this is what the tannins look like over time. So you basically have them in the cap, and they get washed down into the liquid as you go through and you do cap management. These two curves here are essentially the phenolic levels in the cap, and these are in the liquid. So you can see the phenolics in the cap don't change very much, but the liquid increases as you continue your extraction. And this is the prediction from the model. Again, it's not perfect, but you'll see it's very close. And so we can start to use this model to predict other things that I think are interesting points for thinking about as you're thinking about what phenolic profile you want. So one of the things that Conrad did was he looked at how could you maximize skin tannin. So if you wanted to maximize the extraction of tannin coming from the skin, what he found was that actually having pump overs 1 time per day actually gave you more skin extraction than 0 times a day or 8 times a day. And it has to do with getting fresh solvent that will help with the extraction to have a gradient between the skins and the liquid. But it also has to do with the more pump overs you do, the lower temperature your cap is going to be, and you're going to slow down extraction, especially, slow down extraction. We'll talk more about that next slide. And I think this is not obvious to me that an intermediate amount of pump overs will actually give you more skin tannin. Another thing he did was if you wanted to minimize the fraction of C tannin because maybe you don't want bitterness that's associated with C tannin, basically what you need to do in this case, this is percent C tannin, you just need to do more pump overs. So the fewer the pump overs you do, the higher the cap temperature and the more C tannin you're going to extract. Later in the fermentation, but you're going to extract more C tannin. And so you can minimize that by doing more pump overs because you're going to keep the temperature lower in the cap where the extraction is happening. And finally, getting back to that question of whether if you don't pump over, it should be pink or dark red. I think what Conrad showed was actually he did this kind of simulation experiment where he used a 100-liter tank that's squat, short and wide, versus a 100-liter tank that's tall and skinny, and then an 8,000-liter tank that's short and squat versus an 8,000-liter tank that's tall and skinny. And these two in the middle have basically the same liquid height and the same height of liquid underneath the cap. And what he found was the extraction is similar for those. For if you have a larger tank that's tall and skinny and you don't do pump overs, you're going to get a pink wine. If you have the smaller tank that's shorter and squatter, or any tank that's short and squatter, you're more likely to get full extraction without doing pump overs or other cap management. All right, lessons learned from phenolic extraction. Know your cap temperature. It can be as much as 30 degrees Fahrenheit higher than your liquid temperature. It's worth measuring because you could actually cause stuck fermentation by not paying attention to that because your cells will be inactivated or die at high temperature. Punch downs and pump overs will cool the cap, but your jacket is not going to. Your jacket is ineffective at cooling the cap more than an inch or so into the cap. We've made those measurements many times. The larger and wider your tank gets, the greater that effect. So at some point, you probably need external cooling to keep your fermentation at the temperature you want it at. Skin extraction happens early, seed extraction happens later, and it's much more temperature dependent. And cap management is more important as you get to larger volumes and taller, skinnier tanks. So with that, I'd like to acknowledge all the funding that funded all this work over probably 20 years or so, and the students and my colleagues who actually did it. This was a lot of the work that I showed was a collaboration with E&J Gallo, but we also had American Vineyard Foundation funding along the way as well. So with that, I'm going to stop sharing. It looks like there's some questions in the Q&A.
There are two. The first one is, can you speak more specifically about the instability issues caused by excess nitrogen?
Yeah, so it's not necessarily going to be that you will have instability, but if you have something that's growing in your bottle, or you have your bottle is not sterile filtered, and you have microbes in there, they're not going to be able to grow easily without nitrogen. So if you have residual nitrogen from your primary fermentation that's not used up in ml or by any other kind of growth, you're just going to potentially have more chance for problems later on. So that's what I meant by that.
What is your opinion on using a pied de cuvée to kickstart the fermentation?
We haven't done any work on that. So I would suggest maybe asking Nichola in the next talk. I know that sounds like I'm backing out of that question, which I am, but I think asking her might be a better choice. Maybe we can save that for her as well.
Sure. And then one last question that just came in. Any thoughts on submerged cap fermentation?
Yeah, we did try submerged cap fermentation. We basically saw the same extraction as we saw when we controlled the liquid and the cap at the same temperature. So we did that by controlling the liquid with a jacket and the cap temperature with short pump overs. And when we did that, we got basically the results fell right on top of the submerged cap. So I think it's fine. It's maybe harder to do at a larger scale. But you would probably get about the same results, at least from our experiments, about the same results from controlling your liquid temperature and your cap temperature at the same temperature.
And one last question that just came in. Are there ways to remove excess nitrogen if it's not used up in primary or malolactic fermentation?
Probably not easily. So, lots of times if you have a normal amount of nitrogen and you measure that nitrogen during primary fermentation, all of it's going to be used up. Although sometimes at the end of fermentation, you start to see a slight increase, maybe because cells are breaking up and releasing nitrogen. I think having a little bit that's there from the primary fermentation is probably fine. I think you just don't want to be putting in excess amounts of nitrogen. More is not better in that case. Add enough to get through your primary fermentation. If you have questions about the nitrogen utilization rates of your cells, talk to people at your yeast company and ask them what the recommended level is. And again, I would go for that. Don't add a lot of extra just to make sure your fermentation finishes. It will, but I don't think it's a good idea.
Great. Thank you very much. We really appreciate you being here today and hope you're feeling better soon.
Thanks, me too.
All right, it's 10:35 and we wanted to come back, I believe, at 10:40. So we'll take a good 5-minute, maybe 6-minute break, and we will be back with Nichola Hall as our next speaker. Thank you. All right, welcome back, everyone. Our next presenter is Nichola Hall. Thank you, Nichola, for presenting today. Dr. Nichola Hall earned her undergraduate degree in microbial biotechnology and a PhD in yeast physiology and fermentation science from the University of Abertay Dundee in Scotland. She says she fell into the wine industry by accident when she began working her first harvest as the microbiology director for Bronco Wine Company in Ceres, California, back in 2002. She is currently the general manager of the Fermentation and Enology department at Scott Laboratories in Petaluma, California, and has been there since 2009. She is also a co-winemaker for Matthew Bruno Wines in Rutherford, California. In addition, she teaches quality control and analysis in winemaking in UC Davis's continuing and professional education winemaking certificate program. And she's an active member of the American Society for Enology and Viticulture, having served as president in 2016 to 2017. She has, as I mentioned before, she has more than 20 years of experience in the wine industry. So thank you and welcome, Nichola. I think you are muted.
Muted is the best way to have me. So thank you for the invitation and introduction. Harvests go by quick, don't they? So let me share my screen. Initially, it'll be a little awkward as I work out which screen we're going on, but we're going to talk about yeast and yeast choices today. Screen one, initially you'll see my desktop, which is delightful, isn't it? Okay.
Looks like mine, just a few things on there.
Okay, Karen, do you see?
I see your presenter view, not
Okay.
not the complete. Yeah. Yep.
Okay, perfect. So in that case, let me move some things around. So again, good morning, everybody, and thank you so much for joining us today. We're going to chat about yeast and yeast choices. We're just going to jump right into it once I get my cursor on the right page. Okay, so what are we going to talk about today? We're going to talk about why we actually use Saccharomyces species for winemaking. I'm going to very selfishly give you an update on the taxonomic classifications and kind of where we are today. And that's because when winemakers ask me for bianas, I'm like, is that what you actually want and need? Chat a little bit about the species and strain diversity, their impact on wine quality. Once you understand a lot of those things, then you can really appreciate how you can get into the nuances of the different commercial strains and how to be successful in your selection. We'll kind of look at an overview of the different, I call them classes in not the microbiological sense, but the class of different yeast available. If we get to it, we'll review fermentation profiles and overview of what to do when things go wrong, and I will answer that Pirikov question in addition to any others. So if we think about microbial populations and where these things come from, let me get a laser pointer here. There we go. We'll see that, you know, our organisms are associated with the grapes. Depending on the maturity level, we'll have different organisms. When we harvest, that's when we really start to talk about the bacteria and yeast associated with the winemaking process rather than associated with the vineyard and viticultural practices. So our bacteria and our non-sac are entered into the winery from the grapes, from the vineyard. When we're talking about yeast these days, we're talking more and more about these non-Saccharomyces organisms, and non-sac is a colloquial term used to describe yeast other than Saccharomyces. We probably should say other than Sac and Brettanomyces. The non-sac yeast encompasses about 20 different genera and about 700 different species, and to date, we don't have a number of different strains that are available, but they're probably within the tens of thousands. Because we appreciate what our non-sac can do for wine quality, we do now have different commercial strains available, and these commercial strains allow us to exploit the strain differences. and it allows us to exploit their unique attributes and how they can be used to impact wine quality, whether it's using them as antimicrobial agents because they either remove something from the matrix, add something to the matrix, or they just take over the matrix and become the dominant organism in that ecological space. They can work as antioxidants to remove precursors to oxidation, whether it's the oxygen itself, whether it's the metal ions like iron and copper, how they can release or produce aroma compounds, whether it's they've got enzymatic ability to reveal varietal aromas or fermentation derived esters. They could work on body and structure, due to their polysaccharide release, and they can modify acidity due to lactic acid production or other TCA cycle intermediates like succinate or alpha ketoglutarate. So when we're talking about yeast and winemaking today, we can't ignore these organisms, but generally what we do talk about when we're talking about the yeast involved in the winemaking process is Saccharomyces. And Saccharomyces is an organism of the built environment. It's from the brewery, the cidery, the winery, the bakery, the distillery, and it is our organism of choice for winemaking. It has been our organism of choice for a lot of years. If we look at the history of fermented beverages, we'll see back in 1890, the initial concept of inoculating wine with pure cultures was introduced. We haven't really had active dried wine culture since back then, though. The first two strains were available to the wine industry in 1965. In 1973, we added another 4 strains to our portfolio. And back then we're like, well, or I'm told that back then people were like, 6 strains, why do we need 6 strains? But this is when we start to see things like 71B and EC 1118 being released to the industry. Back in the 70s, when we were releasing some of these strains, they were classified very differently than what they were today from a taxonomic viewpoint. As winemakers, you could be aware of the current taxonomic classification, or you can completely ignore it. That's up to you. But when you're calling and asking someone, one of your suppliers, for a specific genus and species, make sure you actually communicate what you need, not what, so that you're both talking the same language. Because Saccharomyces has had some changes over the years. In the old days, pre-1970, we had Saccharomyces ellipsoideus, cerevisiae, pastorianus, bayanus, carlsbergensis, and ovarum. In 1970, due to phenotypic identification, the ellipsoideus and cerevisiae were reclassified to just have cerevisiae. The pastorianus and bayanus were reclassified just to give us bayanus, and carlsbergensis and ovarum were reclassified just to be ovarum. In 1984, they were, through phenotypic identification, they weren't different enough based on the tools we had back then. So we had cerevisiae. In 1996, that's when we had the genome of Saccharomyces isolated, identified and fingerprinted, and there began the molecular era. And in 1998, we had Pastorianus, cerevisiae, and bayanus. So what we've called these organisms over the years has changed, and what we've asked for them or from them over the decades has changed. In the 80s, our main goal was to convert sugar to alcohol and CO2. But we did still have some breakthroughs when we started to discuss yeast sensory contribution. Things like D254 and BDX were released to market. So these cerevisiae, all the same, Saccharomyces cerevisiae, from a taxonomic classification. In the 90s and 2000s, the goal was to convert sugar to ethanol and CO2 with no sensory deviations. And our big breakthrough was when we started to discuss the yeast in terms of their sensory and their textural contribution. And that's when things like RC212, Bourgogne Rouge, and Bourgogne Blanc CY were released to market. So the early 2000s was when I entered into the industry. We are now kind of looking back and still communicating with some people using some of this older terminology, Sangiovese and Bayanuses. But in 2023, kind of what are we using for our taxonomic classifications? We're still using some phenotypic identification. So how does it act? What does it look like under the microscope on a plate? What does it ferment? What does it assimilate? How well does it grow? We're doing a lot more molecular typing, and the molecular techniques that are used have really changed over the past 2 decades. And we're now into the omics realm. So it's not only good enough to know what genes are present in our Saccharomyces, but which genes are active and going towards our metabolite profile or organoleptic qualities of our wine. So where does Bayanus live, right? So when somebody's asking for a Bayanus, Bayanus doesn't really live in the Saccharomyces sensu stricto anymore. When we look at true Saccharomyces strains, we'll see that we have Sangiovese, most closely related to Sangiovese, Paradoxus, Cariocanus. We have some others, including Ubayanus and Uvarum. And Saccharomyces Bayanus is known to be an interspecies hybrid, mainly of Ubayanus and Uvarum, with a little bit of Sangiovese DNA in there. So where we are today, when we're talking about taxonomic classification, we talk about genus being Saccharomyces, species being Sangiovese. So we have a subspecies or a biological variant, and that is a Saccharomyces cerevisiae var Sangiovese, or we have a Sangiovese var Bayanus. The difference really in those two is that the Sangiovese can ferment galactose and the Bayanus cannot. So you'll see some communication on this is a Sangiovese Bayanus cerevisiae, or this is a Sangiovese galactose positive, a Sangiovese galactose negative. That is what that means. So what about those true Bayanus that winemakers still ask for? There really are only a few true that are available commercially. These strains have an exceptionally high nutrient demand, much so than the Sangiovese cerevisiae or Sangiovese Bayanuses. They have a low temperature tolerance and they're relatively low ethanol tolerant, so maybe up to 14%. Okay, so when we're talking about Sangiovese, now we're really talking about the genetic diversity of the organism and what that genetic diversity brings to our winemaking. We know where to look on the genome to determine where the strain-specific diversity is, and we are aware it's on the subtelomeric region. And by doing a lot of the genotyping, so looking at its genetic background, Saccharomyces having 16 chromosomes, 6,000 genes, generally being a diploid, so looking at all this genetic material, we can see that the wine strains are separated from bread, from flour, from rum, cheese, etc. And that means that we've got more than 300 genetically diverse wine yeast strains to choose from today. So those wine yeasts have a very high degree of genetic diversity. They've also are very different in their phenotypic expression. So the phenotype being their genetic background plus their environment. And that's where we see a diversity in the metabolites that are produced. So we'll see the diversity in how much nitrogen they require, how much glycerol they can produce, how much SO2 they can produce, the aroma compounds that they can produce under different conditions, and how much negative compounds they can produce, in this case, hydrogen sulfide. So when we're talking about Saccharomyces, there's a lot of work that goes into studying and understanding the diversity. But the role of Saccharomyces for today's winemaker is generally to have the rapid, complete, and efficient conversion of grape sugar, so glucose and fructose, to ethanol, carbon dioxide, and other minor but very important flavor active molecules without the development of off flavors. So when we're selecting our Saccharomyces and when we are doing all of the isolation characterization work, we're going to look at their technical attributes and their sensory contribution. So when we're talking about their technical attributes, Dr. Block spoke about some of them. I'm going to talk about them again, but in a little bit from a different angle, a little bit more details. But Saccharomyces itself is an organism. We use it because it's able to tolerate or thrive in the harsh conditions of juice and then wine. It can survive in limited oxygen conditions, in SO2 levels, low pH, high sugar, high ethanol concentrations. We use it because it is genetically stable. It has good implantation rates. It has a high, or it should have high viability, so high abilities to grow and high ability to be metabolically active. It has good and predictable fermentation characteristics. We can manage its nutrient needs. We can manage its temperature demands. It's not excessively foamy, and it has a good contribution to wine quality, like good aroma production. It settles well. So from back in the 1970s, when we had 6 strains available, we now will call our strains classic strains. So kind of those neutral fermenters. So we can always call them technical strains. We have specialty strains, so good fermenters that focus on maybe revelation of varietal compounds. We have hybrids, Saccharomyces or Cerevisiae caricanus hybrids, Cerevisiae paradoxus hybrids, these interspecies hybrids. We have blends available, and we have what we call QTL strains. So these are strains that have had the ability to produce H2S through the primary pathway knocked out of their genetic code. So, and that is quantified through quantitative trait loci. So we've come a long way from the mid-1970s, but our attributes and why we use Saccharomyces is really still the same. So let's look at some of the wine yeast characterizations. So yeast ability to withstand alcohol, its tolerance to alcohol. Generally, wine yeast can tolerate between 12 and 18% alcohol, depending on its genetic background. Remember, we're going to use fermentable sugar to calculate how much alcohol will be produced, and Brix is not completely equal to fermentable sugar. Our fermentable sugars being glucose and fructose. Brix, as you know, is grams per 100 mils. Glucose and fructose is grams per liter. So we've got a factor of 10 difference there. And if we look at this ETS guide from Harvest 2019, and we look at different red varietals over 5 vintages, if Brix was equal to glucose and fructose, so 1 Brix would equal 10 grams per liter glu-fru, we wouldn't have anything on above this line. But because it doesn't, we'll see a variation in how much alcohol will be produced by our yeast. So the best way to calculate it is your potential alcohol is equal to glucose plus fructose divided by 16.83. It's a stoichiometric reaction, and yeast are not any more efficient than they used to be, and I'll address that again in a few slides. So yeast relative nitrogen needs, what Dr. Block was discussing. Yeast are classified as low, medium, or high demanding strains. And how we could turn that into a usable number rather than an ambiguous term is if we take our sugar measured in grams per liter, and we're using a low medium strain, you multiply that sugar by 0.75, and that will tell you how many milligrams per liter of nitrogen is required for that strain in that environment. Same for medium nitrogen demanding strains, a little bit more. So the constant we use is 0.9. For high nitrogen demanding strains, it's 1.25. So for example, if we had a 20 or 250 gram per liter juice that we're fermenting, rounding up, we might need between 190 and to over 300 parts nitrogen. So your cells, as David showed, will also respond differently depending on the amount of nitrogens there. And also the type of nitrogen that is there. Dave showed yeast response to inorganic or a monocidal nitrogen. He showed the response to a blended nitrogen nutrient, so ammonia and amino acids. And then if we look at just amino acids again, we have a different response, with the yeast being so much more efficient with the amino. Temperature is a big thing that we are always trying to encourage winemakers to pay attention to. Again, as David showed, it impacts fermentation kinetics, it impacts fermentation outcome because it impacts cell number. What we'll say is a good fermentation rate once you get over the lag phase is about 2 to 4 bricks drop per day. The higher the alcohol, the lower your fermentation temperature should be. The impact of temperature and ethanol on the yeast plasma membrane is similar in the fact that they change the ratio of unsaturated to saturated fatty acids. So instead of having a nice fluid bilayer, we end up with interdigitation, we end up with leaky membranes. The membrane is protecting the cell from the harsh environment. Everything comes into the cell across the membrane and leaves the cell, in theory, through the membrane. So we need that membrane to be intact. When we've got too high temperature, that intact nature of the membrane is compromised up to jeopardized. So 90 to 95 is lethal in the presence of alcohol, as David showed. When you don't have alcohol present, 110, so 40 Celsius, is sublethal, and 115, which is around about 43 Celsius, is actually lethal. So when you're rehydrating your yeast, you actually want to make sure that you're measuring the temperature with a calibrated thermometer. And you never want to ferment at either end of the yeast temperature ranges. You want to kind of be somewhere in the middle. You want to find strains that can work well in your system with your temperature management capabilities. Yeast sensory impact. So as I said, yeast can be neutral. They could enhance varietal characteristics. They could impart esters, and they can work with mouthfeel, among some other things. But these are the big categories we chat about. So a neutral strain is sugar to alcohol CO2 with yeast as your catalyst with no sensory deviations. We're just looking for a good, clean fermentation. Most winemakers are looking for more than that these days. They're generally looking for yeast to be part of their wine style guides. They're trying to use yeast as one part of getting to the final sensory profile. So yeast can have a very positive impact on varietal characteristics, both terpenes and thiols. Terpenes, as you remember, are found mainly in the skins, and these are described as being floral or spicy, citrusy. Sometimes they could be a little resiny. Your thiolic precursor, the 4-MMP in white wines, that we generally describe that as being boxwood or grassy, sometimes even kind of asparagus. In whites. In reds, it's your black fruits and blue fruits, so blackberry, blackcurrant, and blueberry. Whereas your 3-MH precursor is found in the pulp, and that is generally citrusy or tropical. So yeast can impact these major groups of compounds. So their impact on terpenes, the yeast has to have the ability to produce a beta-glycosidase to cleave the terpene from its bound sugar. So different Saccharomyces have, because of the different genetic makeup we discussed, we've got different genes being coded, different genes coded for different enzymes to cleave the monoterpene. So it's going from a non-odoriferous to an odoriferous form. If your terpene is bound to two sugars, if it's not just bound to a glucose, if it's bound to a glucose and a 5-carbon sugar, so that again in the non-odoriferous form, we're not going to pick it up. So the Saccharomyces itself has a little bit of a harder time cleaving off this 5-carbon sugar, and that's where non-Saccharomyces really comes into play. It's also thought that some Saccharomyces can also synthesize these terpenic compounds themselves. Yeast will impact thiols. This has been a really hot topic probably for the past 10, 12 years now. How yeast can cleave these varietal compounds that are bound to amino acids to take them from that non-odoriferous to the odoriferous form. So it's going from something you can't smell to something that you can. And it's all depends on the yeast's ability to, genetic ability, so it can produce the enzymes to cleave the volatile compound from its conjugated form. So what we'll see when we're looking at thiols, the yeast have a huge impact, and the yeast with the IRC7 gene is much more impactful in releasing the 4-MMP, and that's the aroma compound we don't seem to have a huge amount of in our warm climate California fruit, whereas New Zealand has a huge amount in theirs. But just looking to make sure that the yeast have the IRC7 gene doesn't seem to be good enough now. And this is part of the history of the yeast and how our new modern techniques are allowing us to explain and understand their diversity better. So this gene, the IRC7, codes for carbon beta lyase. And the presence of that gene is very highly strain dependent. And we think that about 17% of genes actually have the long form of the gene, and about 56% have a truncated form of the gene. So you've got two lengths of the gene. And if the yeast has the short form of the gene or the truncated form, then it codes for a non-functional protein. So just looking now for the presence of IRC7 isn't good enough. We've got to look for the presence of the gene and making sure it has a functional form of our carbon beta lyase to cleave off our thiols, to increase our floral, increase our tropical notes, increase our citrus notes, or increase our blue and our black fruit notes in reds. So thiols are super interesting just from a scientific perspective these days. As we know, yeast can have a huge impact on fermentation aromas or ester production, right? Is fermentation aromas. Our different yeasts have different abilities to produce different esters. And it is going to be impacted by the yeast genetic background. So does the yeast have the ATF1 and 2 to help produce the acetate esters? Does it have the HT1, etc., to help with the production of ethyl esters? If the yeast doesn't have the genetic background, it can't produce the compounds. We could, if the yeast does have the genetic background, we could manipulate it and exploit it with different means so that we get different compounds. So we can exploit its temperature to have different compounds produce the lipids or the phytosterol content. And we know that we get more esters there when our phytosterol, so our sigmasterol, stigmasterol, campasterol content is about 60 milligrams per liter as opposed to 240. So we want kind of lower lipids, these grape lipids, so a lower solid content to get more fruitiness, and also our nutrient availability as our amino acids kind of feed into the pathway here, get transaminated to your higher alcohols to get your acetate esters. But vitamins have a huge role to play in these pathways as well. So just looking at nitrogen isn't really the whole story. So our Saccharomyces, a huge amount of genetic diversity, 300 different enological strains available. And if we exploit the genotypic and phenotypic traits, we can get full aroma production. Okay, so it's making sure that you have the capacity in your grapes, you extract the compounds, and you use your biotechnological tools, i.e. our yeast, to allow that to be seen in the final product. Yeast also impact mouthfeel, and that is generally due to their manoprotein contribution. So the manoprotein is part of the yeast cell wall. They can vary from strain to strain depending on the length of the mannan chain and depending on the length and composition of the peptide part, right? So it's a manoprotein, so it's the manos with the peptides co-joined. So once they're released, you can get a lot of roundness in the mouth, and they also will help you with things like aroma and colloidal stability. So depending on what manoproteins are in the yeast and when they're released, you'll see different impacts. As we see the polysaccharides per gram of biomass between BM45 and RC212. Your BM45 is going to start releasing them much quicker than your RC212, and you're going to get more of them produced as well. So yeast will impact mouthfeel. They could impact color. They could pick up color on their cell walls, or produce, again, the polysaccharides to complex your polyphenolics so that we have that anthocyanin tannin polysaccharide complex for stability of color. So when you're selecting your strains, right, make sure that you're considering your alcohol tolerance, your relative nitrogen and nutrient needs, the temperature tolerance of the strain. Make sure you can work within those, and the sensory contribution. So we look at the technical as well as the sensory attributes. So what we want to say to you is follow the strain guidelines, please, pretty please, all the pleases in the world. There's a lot of science that goes into the recommendations of the yeast, which means that to give you a recommendation, we've had to do a lot of trials, a lot of experimentation, which means a lot of failure came with that. So when there's a recommendation, it's kind of coming with a lot of work behind it. So kind of let us do the failings so you don't have to in your vintage. So we've got a lot of different wine strains, right? We've got our classic strains, our specialty strains, our hybrids, our non-H2S strains. So what you want to think about when you're working towards your profiles, does my yeast have what it needs genetically? Is the strain capable of doing what I want it to do? And is the environmental conditions conducive to allowing it to do such things? So I want to release thiols. Does my yeast have that ability? Do I have a full copy of the IRC7 gene with the functional protein? That protein is going to be produced at much higher levels at 20 degrees Celsius. So is my environment allowing for my genotypic expression? And then as winemakers, are you giving it what it needs? Are you looking after its temperature, its nutrient needs, its lipid requirements? And all those things will help you with your wine profile goals. So remember that fermentation profiles, fermentation is a function of your total viable biomass, as Dr. Block said, and the rate of sugar utilization of the individual cells. And a lot of things will impact your fermentation rate. So it depends whether you have a normal fermentation, which would be your red line, whether you have a long lag phase, whether you become sluggish, if you're sluggish throughout, or you have this abrupt arrest. Why? Is it because you didn't have enough cells there, or they weren't healthy? You didn't have a good metabolic rate? They weren't very vital? Did you have a nutrient limitation? Do you have ethanol toxicity? Do you have some inhibitors present, whether they're chemical or biological? So SO2 versus high lactic acid bacteria population. Do you have a weird cation imbalance? So you have too much calcium, so you can't get magnesium uptake. Do you have temperature extremes, poor winemaking practices? So it's overly clarified, for example. And remember, these factors can act independently or synergistically. So make sure for you, you know what a normal fermentation profile is, because normal is the goal, right? And then your different inputs will impact that curve. So we really want to make sure we're avoiding problems. So proper yeast choice, proper yeast handling. So you're rehydrating and acclimating as necessary. You're properly nourishing the cells. As David showed, mixing is really important. Temperature management is important, and you control for antagonistic microorganisms. You make sure that you don't have too much bacteria present to inhibit your Saccharomyces. So if you do recognize a problem fermentation, react quickly, know what's going on, both analytically and microbiologically. An adaptive process is simple but effective. Sometimes just adding yeast if you didn't add any and you're only a few % alcohol, adjusting your nutrition to give the yeast what they need when they need it, mixing, getting your yeast back into suspension, adjusting your temperature, combating that microbial antagonism or detoxifying. So kind of know what's going on, go through the steps and get it to dryness quicker. So with that, we've spoke about why we use Saccharomyces cerevisiae in winemaking, and it's due to its compatibility between the yeast and the matrix. The yeast is compatible for the matrix. We have a significant amount of genetic diversity and phenotypic expression. So when you're choosing something, make sure it's going to be successful in your environment. And that successful selection and outcome depends on how much attention is paid to your choice and how much respect is paid to the recommendations. Make sure you're following your fermentation, know what normal looks like, and make sure that you intervene really quickly if things are not looking normal. So with that, thank you. Thank you, Karen and the UC team for the invitation. Very much appreciate it. Thank you all for your time and your attention, and to the scientific community for their dedicated research. So without your dedicated research, we don't learn, we don't improve, and we don't get to brag about the amazing wines we make. So have a fantastic harvest and a safe one. Thank you.
Thank you very much, Nichola. Would you like to answer that question that Dave saved for you? What is your opinion on using a P de Cuvée?
So kind. So I've come out of slide mode. Can you see me?
Yeah, you probably need to stop sharing.
Okay, thank you.
Yep.
So Peter Cove, you can use it to kickstart a fermentation. Your active dried yeast are not adapted to alcohol. So when you're introducing them to an alcoholic environment above a few %, I used to say 8, but I'm kind of coming down to more than 5%. You want to do some sort of acclimatization, and a P de Cove is a type of acclimatization. So you do it for restarts, we do a P de Cove, and we'll do it for sparkling wine as tirage.
All right, there is one more question here, and it's, has Baianus always been understood to have low ethanol tolerance and high nutrition demand? I understood the opposites, i.e. champagne secondary fermentation.
That's because you are living in the 1980s, and this is why it's kind of a pet peeve as a microbiologist. In the old days, that's what we used to say, right? But sparkling strain is a Cerevisiae Baianus. It's not a true Baianus. True Baianus, there's only 3 or 4 commercial strains, and you did understand correctly. They are low in their nutrient demand, low in their alcohol tolerance and very, very high in their nutrient demand. So that's why when somebody asks for a Baianus, I'm like, what do you actually want and what do you need? It is a kind of a misunderstanding that's been perpetuated through the decades in our industry. So thank you for asking.
Great. Thank you very much.
You bet.
We appreciate being here
Happy
today.
to participate. Thank you. We'll see you all soon. Bye.
Great, thanks. All right, our next presenter is Anita Oberholster, and I'll read a brief introduction while she is getting her presentation shared. Anita is a vice chair and a professor of cooperative extension and enology here at UC Davis, and she joined the department in 2011. Prior to UC Davis, she was a researcher and technical officer in the Department of Viticulture Enology at Stellenbosch University in South Africa. She received her PhD in wine sciences at the University of Adelaide in Australia, and a BS honors degree in chemistry and a BS in chemistry and biochemistry from Stellenbosch University, also in South Africa. In her current position, she focuses on continuing education for the grape and wine industry, while her research program concentrates on current issues in the grape and wine industry. Her research topics include the impact of climate on grape ripening and phenolic development, grapevine red blotch disease, and smoke exposure in the vineyard, which you're going to hear about today. Another major focus for Dr. Oberholster is the impact of different winemaking techniques on wine composition and quality. Anita has received a number of awards recently, including the Extension Distinction Award from the American Society of Enology and Viticulture, the Distinguished Service Award from the UC Davis Academic Federation, and the California Association of Wine Grape Growers selected Anita as the Leader of the Year in 2022. And just this past year, she was one of 3 researchers working on the impact of smoke exposure that received the Innovators of the Year award from wine enthusiasts. And I will tell you, she just received 2 more awards in the first half of this year, but since we're running a little late, I'm just going to let her start her presentation. Thank you, Anita, for joining us this morning.
Well, thank you very much for that introduction, Karen. Is my sound okay?
Yep.
Okay, so let's jump in as we're already a little bit late, and I'm known for talking too long when I talk about smoke because there's so much to say. Good morning, everyone. So I'm going to focus this presentation mostly on the grapes, but at question times, question time, ask me anything about anything. So quickly, just about the science, and my thing doesn't want to advance. Let me just get this. There you go. So I like to give the background science. I'm going to give it in a very, very short period of time. So when we have wildfire burning, we have a lot of vegetation burning. And for instance, wood, 15 to 25% of wood contains lignin. Now that's what you see here on the left side of the screen. Now, when lignin thermally degrades at high temperature, it can release massive amounts of those ring structures. So if you've seen the green, you have what we call volatile phenols. A volatile phenol is something with a ring structure and a hydroxyl group on it. And then it can have various substitutions. So there's a methoxy group. This specific volatile phenol is galco. So everybody's familiar with galco now. Now, if you have a wildfire and you have a massive amount of volatile phenols being released, and you're unlucky to have a vineyard nearby, those volatile phenols can disperse in the air to that vineyard and cause issues. It can also absorb onto debris in the air and actually get distributed on the debris. Now, if you have a vineyard close by and you have fresh smoke, it can absorb onto the berry and inside the berry, but it will also absorb onto the leaves, heavily onto the leaves as well. So it really goes into both. Now, we think this is part of the defense mechanism of the plant. It also increases the solubility of these compounds. They get glycosylated by glycosyltransferase enzymes inside the berry. Now, what this means is various sugars can get attached to the range of volatile phenols being absorbed into the berry. It can be 1 sugar, 2 sugars, 3 sugars. So you now have this whole combo of different combinations of sugars that can absorb onto these volatile phenols. So now we're looking for a very large group of compounds. Now, the Australians has identified these 7 free volatile phenols to be associated with what we call smoke taint character in wine. I analyzed a few more, and I found 2 that also correlate really well. You don't really need it for prediction. These ones are pretty good, but we also analyzed for ethyl glycol and for phenol itself. Now, these compounds and their glycosylated precursors are naturally present in grapes. So this is very important to understand. Usually at very low levels, but I have seen vineyards with high levels of natural baseline. So it can vary. We know it varies by variety, but it can also vary by site. If you think about this a little bit, if you think of the quality of air, the quality of air is not as good next to a massive highway or very close to a town than it would be on a mountaintop, for instance. So it would make sense that the same variety growing in different areas, just even in Napa, may have natural different baseline values, because it's not just due to what gets biosynthesized in the grapes, it's also what they will absorb in the atmosphere. Now, these grapes never develop smoke taint without actually having exposed to for smoke from a fire. In addition to that, grapes can handle the natural mounds that they get introduced to. Now, I really want to talk about this slide. I stole this from Tom Collins the first time I saw it because I think it's a very good visualization. Now, I understand that for crop insurance, you only need glycol and 4-methyl glycol in the free volatile phenol form. But we're really advocating for people to look at both free and bound, because this is a situation you can have. It's not always the same. Researchers have looked, we do not see a consistent ratio between bound and free. So you can't measure your free and say, oh, now I know how much bound I'm going to have. In some years, we see very little free in the grapes, and there's massive amount of bound because everything that grape was exposed to actually absorbed into the berry and got glycosylated. So this is really important, that you really need to look at both. So quickly looking at the methodology, we have quite a few labs that will give you the free volatile phenols and will give you the whole range, those 7 compounds. And you know, this is a situation where the predictive power isn't 100%. So you really want to measure everything you can to enhance your chances of being able to see what's going on in your grapes. And then they will give you a measure of total. So how do they do the total free volatile phenols? Well, it's usually some kind of acid hydrolysis process. Now, if you look at here, this is the conditions many of us use. 100 degrees Celsius, pH 1, you need strong sulfuric or acetic acid or hydrochloric acid, actually, to get down to pH 1 for 1 to 4 hours to try and break most of these bound into the free form. And it's not 100% that you get, but that gives you an idea of everything is bound. So it's more a qualitative test. It will give you an indication that everything that may potentially be freed during the lifetime of your wine. So if you had smoke exposure, you will see hundreds, okay? If levels are lower, then perhaps the smoke impact wasn't that bad. And we're working on baseline values for this. We're going into a 3rd season. So hopefully by next year, we'll be able to give baseline for at least California. Now you get tests that actually analyze the individual bound. So they analyze for 6 of these individual bound compounds. Now, obviously, there's way more, but Australians have shown good correlation to smoke taint, expression in sensory, and my own work has shown the same using these 6 compounds. So you can either get an idea of everything or have a very accurate measurement for 6. Whichever one you use, it's up to you. What is important is you can't jump between methods because they don't give you the same value. In my experience, they correlate, but they don't give you the same value. So whatever lab you start off with, you want to stick with it so that you can compare the values with each other as your wine or your grapes evolve, or from year to year, so that you can get an idea what's normal or baseline for your grapes and what is a normal baseline for your grapes when they actually make impacted wines. Something else I want to mention. So how accredited labs work is before they run a set of samples, they will have a QC sample, a quality control samples, with known amount of the compounds they're analyzing. And if those values come back within 5% of what they need to be, their instrument on that day is correct and they can run the samples. But what that means is there's acceptable variability of about 5%. That means that these 3 numbers, now this is for wine, microgram per liter, for grapes, it would be microgram per kilogram. For these 3 values, has the same value because that's all within 5%. So when you look at your data, remember this 5%. 5% variability could just be laboratory variability and not actual changes in your wine or in your grapes. So this is probably the question I get the most. What is my risk? And it's one of the most difficult questions to answer. So we know the fresher the smoke, the greater the risk, because the fresher the smoke, more of the volatile phenols are actually present in that smoke because you can't see them. Now, air quality indexes, which a lot of us look at because there's sensors everywhere, is not a direct correlation to smoke taint risk. Because air quality index have things for human health. What is the concern for human health? So it has particulate matter in there and ozone as part of the calculation. However, when that smoke is really fresh, I do think there's somewhat of correlation there. But as that smoke ages, that correlation actually diverges and it doesn't correlate anymore. The thing why this is happening is that volatile phenols breaks down actually pretty quickly in the atmosphere while it's in the gas phase. Now, we said they can absorb onto your particulate matter, and we do not really know, or droplets, what the stability is in that. We currently have a multi-state grant collaborating with atmospheric scientists, and they are actually investigating this phenomena so that we have a better idea of how long volatile phenols actually stay in the air under different atmospheric conditions. So at this stage, we go, if that smoke is less than 24 hours, then you probably have an issue and you may have to test your grapes. So this multi-site grant that I was talking about, so we were trying to get 21 sites up and running in California in last year. What was that? 2022. We are still missing in a few sites. And we're trying to get all of them up and running, at least for this site, to get good background measurements. We're hoping for absolutely no fires. We don't need fires. We want more background and data. You can see it measures a lot of different gases, as well as volatile organic compounds that captures some of the volatile phenols we're looking at. And then we look at particulate matter, 1, 2.5, and 10 micron. We added 1. We're trying to see if the very small particulate matter correlates longer with the VOCs, and we can use that as a proxy, because then that will make life much easier. We're taking air samples to get background air samples as well, and correlate that with the VOCs of the VOC sensor in there, grape samples, and doing microfermentation. All this data is going to the atmospheric scientists so that they can do risk assessment and modeling so that we can in the future do better predictions of what vineyard is safe or what vineyard is not safe. This stage, we can't say safe distance. Just if you think of a wildfire, you sometimes have strong air currents in a direct direction, and say a northern direction. There's a vineyard 15 miles away, but it gets that whole column of smoke within 6-4 hours. Then you have a vineyard that is 10 miles away from the fire, but opposite direction of air current, get no smoke. So this is why a safe distance is a very hard question to answer. This is another, so that was thingy sensors in the photo. These are agrology sensors. I work with them as well. Always good to have a backup system. What I want to show you here, this is all background. Okay. So this is 4 different sounds, site, all background. The VOCs are the green lines in milligram per cubic meter. And then your particulate matter is in microgram per cubic meter. And what I want to show you is that VOCs and particulate matter does not correlate, and that different sites do not have the same amount of VOCs. So it's really very important for us to have background data for these sites before there is a smoke event there that we can look at the difference. Now, the VOC levels for background really varies around very little to about 1 milligram per cubic meter. So we did a smoke taint experiment just a couple of weeks ago. I was trying to get the PM data on here, but it's very complex and I'm still struggling with it. But this is the VOC data. And what you can see is baseline is really low, and 10 o'clock we put the smokers on. We put the smokers on in the tent. There was nothing in the tent except for these sensors, and we went. So this is in milligram per cubic meter. We're up to 9 out of 10, and then it settles down because what it does is all those VOCs will absorb on everything that's in there. The thick plastic of the tent is made and everything else. And of course, it started spreading. When that smoke came in, it blew in the middle where the sensor was. And then we have industrial fans that spray it out. So when it spread out to the tent, it stabilized a little bit. But the average here is between 3.5.22 particulate matter, where in the stable environment, it was at less than 0.5. So you can see that increase in VOCs with smoke. Now, this is an old study, which I like to reiterate because there still seemed to be some confusion about this study. So this is a very cool study that they did in Australia more than 10 years ago, where they intentionally over 3 seasons smoked Merlot vines at different development stages, right? And they determined that it was the most susceptible to smoke at variation. And then that susceptibility went a little bit down to harvest, probably because enzyme activity in the grapes also go a little down with harvest and less got bound. But the fact is, they never said that it's not susceptible to smoke if it's less than variation. It's just more susceptible because the skin is softening and it's a bigger berry, bigger surface area. We have now instances from Australia where they get wildfires much earlier, where grapevines are heavily impacted, and grapes were heavily impacted and made smoke-tainted wines, and they were exposed at pea size. It may just take a little bit more of smoke, but you're not, they don't have Kevlar around them when they're hard and green. They can still absorb volatile phenols. The good news is they only absorb onto the leaves and onto the grapes. So there's no carryover. I've used grape wines that I've intentionally smoked the next year for non-smoke tainted issues or for research with no issues whatsoever. So people ask me about this. So what about washing grapes? So washing grapes doesn't work. These things doesn't sit in the outside. It's inside. Yes, you can wash your grape to remove dirt and other things from it, but it's not going to remove the volatile phenols. In labs, researchers have used even organic solvents and it didn't do any difference. Now, what about ash? So we collected ash in 2020 from the vineyard, analyzed it in the lab. Remember, this is under 60 degrees heat in the SPME valve. We saw the ash release volatile phenols up to a week. Now, but you have to remember, this is in a little vial. In a vineyard, big atmosphere with no smoke, you're releasing these volatile phenols at low amounts. Is there enough to actually go into the grapes itself and cause a difference? We did try to wash off ash from grapes. I don't think it was majorly successful. Sometimes it forces it into the grape bunch. Wet ash actually gives off more volatile phenols than dry ash, then it's lying on the floor. I don't think it was, I don't think it was doing much. I know some people use blowers to blow it off. I don't think you will remove everything because blowers, grapes themselves get sticky, and I find the ash sticks to it. What we do know is, if possible, you don't want mold included, specifically leaves. The leaves have a lot of volatile phenols absorbed onto them. And if they're in the fermentation, it will desorb from those leaves. So if you have to do machine harvesting, see if you can remove as many of the leaves as possible. So we did this little experiment in 21 and 22. Now in 21, we made ash from pine sticks. And you can see how much we threw up there. 1 gram per like 100-gram bunch. That was a guesstimate. Put them in nut bags and we harvested them over time. So even the 7-day exposure to this fresh ash, we didn't find any real differences in that ash. And that ash in laboratory conditions were giving off volatile phenols. So we didn't see any impact. We also did small-scale fermentations, not on the same ash. We got other grapes in the same amount of ash was added to the small ferments. So you can basically see this is with ash, this is without ash. You can see the clear difference. That's a 10 gram per liter. That's massive. Now, the person that was supposed to do this left the lab, so I ended up having to do some of this. And so we only analyzed the really, really fresh ash. And the really fresh ash did increase the amount of volatile phenols in the wine. So we didn't have enough to taste it, but there was a massive increase. I don't think you would have seen anything 7-day-old ash because under laboratory conditions, those ash was not even giving anything off when you extract it or did SPMing. 22, we thought, okay, but does that relate to natural vegetation? So we collected natural vegetation that's been cleaned out around vineyards, and we burned that. The problem is that ash wasn't giving off any volatile phenols or extract any volatile phenols. So I don't think we're going to see much. We're analyzing the wines now. I think that it was burning too hot and all the volatile phenols are gone. So we may have to try it again this year. But I wanted to use natural vegetation, but it doesn't help that after it burned out, there's nothing there. Smoldering ash seems to be better, but we're trying to put this ash through a mash so that they're all the same particulate size. So we have all the same surface area for research purposes, which means it takes us a few hours before that ash, 4 hours to get that ash ready. And I think we lost the volatile phenols in 22 during that process. So what I would recommend is, I would say, if you have old ash, I don't care much about it. If it's fresh ash in a winery and you are capable to rinse those grapes, then perhaps do it. So barrier spray trials. I'm going to include what we've done and what others have done. So Cowland first, or surround. So in Australia, they saw some efficacy when they sprayed it in Merlot, but not on Sauvignon Blanc or Chardonnay. Now they say the variability is probably because of the canopy structure, because you get 30 to 40% coverage at most in the vineyard conditions. And they thought perhaps they got a little bit more on the Merlot than they got on the Sauvignon Blanc and Chardonnay. Now, we did in 2021 a study where we actually dipped the bunches. And when we did that with Cowland, we got about a 30% decrease in the amount of volatile phenol absorption. But when we actually, you'll see that in the greenhouse, we covered that with plastic and we smoked these grapes. There we're harvesting the grapes. And we sprayed that with a backpack sprayer. Only the bunchon and some of the bunch and the canopy. And I can tell you, we saw very little efficacy. We saw better efficacy when it was the canopy and the bunchon, but even with both, it was 10% reduction only, and it wasn't significant because there's a lot of noise in vineyard data. So something, but really not a lot. Biofilm Parka, Canada did the study on Pinot Noir, intentional smoking, so like a 300% reduction in volatile phenol absorption. But when they read the study, they didn't see anything. It didn't actually make it good. So they're like, oops, they said, oh, well, the second time around, they did this under moist conditions. There was a lot of water and moisture in the air, and I think the Parka never got dry because it actually made it worse. Now, both Australia and I looked at Parka, and in both cases, it absorbed more volatile phenols and resulted more volatile phenols in the grapes. And mine was definitely dry, and we rinse off the barrier before we analyze the grapes. So I'm concerned about using Parka. I'm also looking at a different compound from EMP Polymer, GM3E. We applied it exactly like the Cowland 1%, and it shows exactly the same efficiency, basically at this point, as Cowland or surround. A little bit better if you do both the canopy and the bunchon, but it's around 10%. It wasn't significant. We're going to redo it this year, doing a larger stretch, less variables, more vineyards, more vines, that hopefully would decrease the variability, and so that we have enough for sensory to see if it actually makes a sensory difference. Timing of application is also very important. So we applied these things 10 and 21 days before the smoke, smoked it, wait 21 days and harvested it, and we washed everything. We do have an unwashed portion as well, but the unwashed portion the students are currently analyzing. So I don't know what the data showed. Tom Collins found that you have to wash the grapes because it seems like the barrier spray is only sitting out in the outside of the berry. It's more a barrier absorbing the volatile phenols itself, and it stays there. It seems to be stable. So if you make wine, it can desorb from this barrier spray into your wine. So just a summary for the vineyard. Remove vegetation if you can. For winemaking, if you know you have smoke impact, we recommend hand picking just because a lot of these compounds are in the skins. If you start breaking the skins, it will go into the juice. It's less important for red grapes if you're going to make a red wine. For white grapes, obviously, there's the option that the winemaker can have little skin contact and get more flavor from yeast selection. I know this is not always possible, but remove leaves if you can. At this stage, we don't recommend removing the canopy. We know, or removing leaves. We know that the leaves absorb a lot of the volatile phenols, and it can actually help protect your grapes during a smoke event. The research show very limited translocation from the leaves to the berries. It's not zero. I currently have a project looking at this some more to see how much that translocation really is. Because we've seen, we spray on the canopy and the berries, we got a little bit, it wasn't significant, but a little bit more protection. So there is a little bit of translocation taking place. The problem is if you pull leaves after a smoke event, you're going to have an issue of sunburn potentially. And if there's another smoke event, you have no protection for your grapes. So this is why we really do not recommend. If you know you're going to harvest very soon and not immediate, perhaps that's an option. Berry sprays are preventative. Do not try and spray them. Here comes some smoke, let's spray them. If you have anything wet on your berry, it seems to increase the surface area, getting more of the volatile phenols absorbed, or it's some other mechanism, but it seems to make it worse if you have something wet on your berry. Definitely do not spray anything on your grapes while there's smoking conditions. You have to wait until the smoke clears, even for fungicides or pesticides or things. So what's our current industry recommendations? People ask me when to take a sample. Your best sample that relates the best to the wine you're going to make is always going to be one that you take the closest to harvest. Now, here's the problem, obviously. The closer we get to harvest, the higher our risk for a smoke event. So to get away from that, I really recommend getting a chest freezer or any freezer and take samples from variation every couple of weeks. Store that at -20 degrees. Now, at the minimum, you need about 300 berries. Now, it needs to be representative of your vineyard block. And if you're going to keep samples, they're much easier to keep them intact in clusters. So I actually prefer to get cluster samples, one per vine, that really represent that the whole vineyard block. And make sure top, middle, bottom clusters, inside, outside of the canopy. There's within one bunch more variability sometimes than between vineyards. If you take those to a lab for analysis, always keep some of your sample. You never know when you have to go back. So many people contact me now for legal reasons. Want me to look at their data, and they only have alcohol and methyl alcohol. I can do nothing with that. I need both the free and bound extensive profiles to be able to tell you where that smoke happened, or perhaps have a better chance of telling you, was the smoke exposure in the vineyard? Was the smoke exposure in the winery? Where did this happen? What is going on? Just alcohol for methyl alcohol tells me nothing. Remember, as soon as that wine see any kind of oak, even oak chips during fermentation, your free numbers totally compromise because they release a large amount of free volatile phenols into your wine. I can look at those numbers. Then doing bucket fermentations. So why would we recommend that you do bucket fermentations? So here's the thing. During winemaking, at least about 20 to 25% of that bound can get released into the free form. And we know currently for crop insurance, you need only those two numbers of the free. So if you had a large amount of bound because you actually were smoke impacted, some of that will get released and you'll see it in the free number. And that free number will reflect better the risk of your grapes, developing making smoke-tainted wines. So we have a protocol, West Coast Smoke Exposure Task Force developed a protocol based on the Australian protocol. I made a video about it. I had no script, so be nice to me, but it gives you an idea of how to do this at home. And also the other thing to remember is these small scale fermentations, you can actually taste them. And you will be able to taste smoke taint even if a lab can't pick it up. The most important thing there is 20 to 25% of people can't taste smoke taint. We think they don't have the enzymes in their saliva that can release these compounds in mouth, because that very characteristic ashy taste that you get, that is actually, we think the bound being released in your mouth really contributes to that character significantly. That is just a hypothesis at this point in time. So make sure whoever tastes your wine can actually taste it, and they need to taste it blind, and it needs to be more than one person. Okay, and really don't worry about the aromas. Focus about the ashtray. The aromas can come from many things. It can be many other aroma or flavors can cause that. Your wine also needs to be dry. Sugar can really hide that ashy taste for a while, so make sure your ferments are dry. You can do smaller ferments, not bucket ferments. We do the microscale, which is the coffee presses. Tom Collins has what he calls the nanoscale, which is in jars. The thing there is, I still take the same amount of huge sample. I take clusters in a 2-gallon bag, destem them all, mix them all up, and take something out because it needs to be representative. So the smaller you go, the less grapes you need, but please still get it from a representative sample, or it may not reflect what you need to see. I think I went backwards. Okay, so that's my story. Questions. I actually put something about thiophenols there because I didn't cover this in this presentation, and I thought somebody may ask me about thiophenols. So, you know, Elizabeth and Tom Collins found these compounds. So they looked at smoked wines, and Elizabeth had a method looking at phenols for another project. Actually found that there are thiophenols in these grapes. So we've known for a while that when the grapes gets exposed to smoke and we get glycosylation happening, that there's this dip where it goes somewhere else, where we don't see the free just disappears, and we don't see the bound. And we've also known that we don't get mass balance. So the amount of free that gets, we can actually analyze in the bound form is not everything. Now that can be because there's a lot of glycosides we're still not finding. That's totally true. We don't look for every glycoside, or our methods may be missing it. But we've always suspected there are other precursors as well. So the thing with thiophenols is they're present at the nanogram per kilogram level in grapes, or nanogram per kilogram level, nanogram per liter level in wines, with your volatile phenols are 1,000 times higher microgram per liter. So they're very low amounts. So you need a GC-MS-MS or LC-MS-MS to look at them. They are have very low odor threshold levels, so you can smell them at that very low levels. Now, currently looking at only the volatile phenols and the bound volatile phenols, I've talked to the Australians about this as well. We get very good prediction and very good correlations with smoke taint expression with sensory panels. So how important the thiophenols are for prediction, we don't know. It's a very cool scientific finding, but we currently don't know if it's really needed to predict smoke taint risk. We're currently trying to put these methods in my lab. Elizabeth Tomasino has one, and she is analyzing some of our samples from last year, and we will see what we find. So I will keep you posted, but at this point in time, there's no commercial lab that analyzes these samples, these compounds, and we don't know how important they are for risk. So go to the lab that you've been using and just get the full extension of panels of the free and bound wherever you go. Thank you.
Thank you very much, Anita. It looks like Caroline put a link to the small scale fermentation video in the chat. So if anyone is interested in watching that video, it is very informative. And Anita's right. It was our first attempt at doing a video, and it was, but I think it's very useful and gives you the tools that you need. So if you need it, it is there in the chat. It doesn't look like we have any. Oh, Christopher has raised a hand. Caroline, can you allow him? Let's see. Can you allow him to speak?
I can. One moment. Let's try this. Oh, no, he lowered his hand. Raise your hand again, Christopher, if you want to speak. If not, we'll assume that.
No,
was just-
he wrote, sorry,
Ah,
it was a mistake
there you
in the
go.
chat, so that's okay. So we do have one question here. Could you explain the difference between free volatile phenols and bound volatile phenols?
Okay, so free volatile phenols are the free hydroxyl groups, so they're not bound to anything. As soon as they're bound to anything, we call them bound or precursors, because if they're bound, and the bound that we are analyzing for are the bound to sugars. So they're so they're not volatile anymore, but they're very soluble in a water medium. So they very quickly extract with the juice into your fermenting must. And the problem why we're concerned about the bound, even though they're not volatile, is during the lifetime of the wine, they can slowly hydrolyze, and you can actually taste them, because most people have enzymes in their saliva that if you drink a wine, that bound actually get released in your mouth. And so retronasally, you can actually observe them in their volatile form, and that contributes to that awful ashtray character that's so significant about smoked wine. So bound is when it's attached to something that makes it non-volatile, but it has the potential to become volatile and contribute to smoked wine.
Great, thank you very much, Anita, and thank you for speaking today. I would again like to thank all of our speakers for sharing their time and expertise today, and Caroline for logistical help. I also want to thank our extension partners for their continued support. And I want to thank all of you, the attendees, and we hope that you all have uneventful and successful harvests. Caroline did put a survey into the chat and also mentioned that we are going to be following up with sending you all a link to the recording, but also when we send you the recording, we will also send a link to the survey. So if you don't catch it now in the chat, we'll be sending you the survey in a few days. And with that, thank you all very much. We really appreciate your being here. We appreciate all the speakers. And like I said, please have uneventful, successful harvests.
Yes, we're hopeful. Fingers crossed. Okay.